First of all, I thank Harmon for inviting me here, and as Harmon said, this is right around the corner somewhere, so you should avail yourselves to all the great sculpture here. And so what I'm going to talk to you about today is first I'm going to give you my little take on statistical methods for functional genomics, and that basically is my take is that this is the way biologists grapple with large amounts of data. That's what these statistical methods do, and I'm going to, after I give you a little perspective, I'm going to talk to you about kind of illustrations of this. I'm going to talk to you about models, networks, and some practical tools. And I think you have all my slides, but you can, all the slides I have are freely downloadable, and you're welcome to just do what you want with them. So, first of all, I wanted to start off with just a little bit of perspective on, and this is a little bit of context. I know you guys have been hearing a lot about the different methods, but I just wanted to give you a little perspective on things. And so the perspective I wanted to give you was from this fairly well-known, he's a database scientist. His name is Jim Gray, and he actually had this idea of these paradigms of science. He published this a number of years ago, and his big thing was something called the fourth paradigm. And he sort of talked about how initially science was a very kind of empirical thing, and then we kind of developed this idea, which is actually fairly recent in the scientific world, of a kind of theoretical branch where we have models and so forth. And then in the last few decades, we have the advent of computers and computational research. And now we have this new thing that he calls data exploration, or e-science. That's the term he used, e-science. But actually what I like to do is to really basically describe this third paradigm, the computational branch, essentially simulation, okay? This is where people try to simulate things on the computer, and I like to call his e-science essentially data mining. Okay, that's just, and and I think that these two kind of paradigms really give you a sense of how people approach computing and science in general. And the simulation paradigm, this is, as Harmon was saying, I come from a little more physical background, and this is very much the physical mind. How do you predict a rocket trajectory? How do you simulate Newton's laws, and so on and so forth? And the data mining perspective is fairly new, and it's what you're really talking about here. And it's really how we use lots of statistical relationships and also databases and so forth to discover relationships that are kind of hidden in data. So Jim Gray, he died in this kind of freak accident in about 2007. And it's about 5-6 years ago. And, it's kind of interesting. After he died, he was so famous that people put a book out. That book, the book I showed you here, was posthumously published, okay? And then after that, what happened after that? Well, since 2007, there was this steady growth in computation, just more and more computing and whatnot, and even bigger and bigger data sets. And suddenly, around maybe a year or two ago, suddenly people started to have this concept of what they call big data. So this is literally a Google Trends slide that shows the rise of that term, big data. And now, mostly when people talk about big data you hear about in the world, they're talking not about biology, they're talking about just the commercial world. They're talking about just all the information that Google and Facebook and whatnot is compiling, and all the information and financial transactions and whatnot. And this is supposedly a new phenomenon that's really a big thing now. And in fact, I was really surprised. Last year, I gave a talk about this, and when I did, I noticed that last year in the October issue of the Harvard Business Review, they called data scientist the sexiest job of the 21st century. So data scientist is really the term for the person who grapples with this big data. So this is a kind of buzzword in the current world, but you probably also know that big data is a big thing in science. And actually, a lot of the ideas of how to handle big data really developed from science. And I'm going to place what we're going to talk about today within this context. And we see it a lot, of course, in science. We're going to be talking about biology, and we're going to talk about DNA sequencing, but you see it in many other contexts. And I want to start very broad. You see it in neuroscience, you see it in higher physics, astronomy, and so on and so forth. And both science and nature have had these two big data issues recently where they talked about this phenomenon. Now, the big data explosion in biology, to some degree or large degree is really driven by sequencing. You've probably seen this type of graph before. It just shows kind of the hyper-exponential fall in sequencing prices over time. This dot line is Moore's law, which is a straight line on the log graph, and it shows how sequencing is getting cheaper and cheaper. And around 2007, you probably all know this, we moved into this amazing era of next-gen sequencing, where you can sequence everything for virtually nothing. And actually, I published an article a few years ago where I kind of hypothesized that, well, in the future experiments, the sequencing cost of the experiment fraction is essentially going to go to zero. So the act of sequencing itself will cost nothing. And that means that all the cost in sequencing experiments will basically come down to either people doing analysis, or, which is actually as important, is people procuring very valuable samples and important samples. And so that, the future of genomics will really come down to really interesting samples, pre-sequencing, and lots of number crunching. So I want to give you a little perspective on how we think about big data, and I like to make this distinction just to place biology and how we think about it. So there's one way of thinking about big data where you don't really care so much about the structure of the data, and I like to think about like Google search here. When you do a Google search, you're dealing with a large amount of data there, but you don't really think about the structure of that data. You just want the answer to your question. I put the query in, get answer out, right? And the other type of situation is more of like called the Google Maps type of situation, where you want this panoramic view of this huge amount of data. And that's, as I'm going to argue, that's kind of what we want a lot in biology, right? We want to do the search, but we also really want this Google Maps, okay? And I'm going to give you a sense of that. So first, a little bit more of what we see in science. In one of the disciplines that people talk about in science that has to do with a large amount of data is particle physics. And this picture here is sort of a picture that represents this amazing discovery that they had recently, where they discovered the Higgs boson. You probably all heard about this. And the Higgs boson is interesting. They have to deal with just truly gargantuan amounts of information, petabytes of information to find. But they're not really interested in the structure of that information. All they want to do is find a few key particle traces that substantiate the existence of this particle. So it's very much like the Google perspective. It's like go through petabytes and find the few bits of information that you can argue that find the Higgs boson. This is some representation to how many times you find it. You find them just one every billion events or something. Now, a completely different perspective is this is a picture from the Sloan Digital Sky Survey, which is another big data project in science, and this is where they're making a map of the sky. And here, of course, the structure of the data is really explicit, but it's also very easy to understand. In a sense, it's trivial. What is the structure of the data? It is the cosmos. It's what, when you look out in the sky, there's a natural structure that we're very, very accustomed to. Another type of interesting type of structure that you see is people often, they mine human activity. So all of us doing our thing every day generate lots of data. And people have coined this term data exhaust to describe our emanations of data. And people actually mine this. So there's these people who mine Twitter for the global mood, and they've determined that people globally are happy at certain times in the day and whatnot. No, but kid you not, and this is funny as a scene. This is published in Science, which I find quite interesting. And then here, this is an interesting article that was in the New York Times by this very famous scientist named Stephen Wolfram, who long before the advent of all this big data and whatnot, he was compiling every last bit of information about himself. And he's been charting every single keystroke he has done for the last 23 years. And he's graphed them on his blog, and you can tell on average when he's at his computer, on average he talks on the phone and whatnot. I'm not sure it's scientifically interesting, but it's extremely entertaining, and I would recommend reading this. And I just wanted to say we're very interested in this type of large-scale activity mining, and we've just done parenthetically. You can look at publication in biology as a kind of information diffusion type of process, very much like the way people, tweet, or the way they behave in large scale, in terms of large scale human activity. And you can try to make models for how ideas we have in biology, for instance, ideas of how to process next-gen sequencing data or discoveries we have of how they propagate through our community. And you can analyze this. We have these multiple models to do this. So the other example I'm going to show you, which is a little bit more to home, is one project that I've been involved in biology that really it's trying to do the same thing as the Sloan Digital Sky Survey. It's trying to create a huge map of the genome, and this is a project called ENCODE. Now the problem is that ENCODE faces is that we don't have an immediate intuitive representation for what that map is. There's the picture of the chromosomes, which you're probably all used to, but there really isn't anything that's as intuitive as looking out in the sky and seeing the Big Dipper and seeing the moon and stuff like that. So, but ENCODE is trying to give us that map of the genome, and that's the annotation, by kind of layering a lot of data together. So I'm going to talk a little bit about this type of presentation today. And one of the key ideas, I think, in dealing with big data and trying to create a structure to it is really trying to there's statistically mined, but a very big part of it is how do you organize it? How do you take all of this information and provide a coherent structure to it? And so one of the useful ideas here is kind of a hierarchical presentation. And so last year there was a sort of rollout of a lot of this human genome annotation, and people spent a lot of time thinking about how to organize this. And so you can, if you just think about it in a very hierarchical way, there's the actual raw data from the genome, which is all the reads that you get from the next generation sequencing. And we're going to be talking about this in a second. And they get summarized into signal tracks. And then there's higher level summaries. We're going to talk about them in a second, that are, where transcription factors bind, or how they come into networks, or how much a gene is expressed. And then on the very top of this is people write papers. They write papers that have human understandable ideas, and they have comprehensible figures. And you can think of this as kind of hierarchy, and it almost meant to be kind of explored from you read the paper and you go to these data sets and so forth. And you'd be kind of stupid to start off with literally the terabytes of data at the bottom. It's really meant to be explored in a certain direction. And it's also very important, if you think about things in this hierarchical structure, you want to have linkages between all the different parts. So you want to be able to look at a picture in a paper and actually understand how that gets transformed into the data in some database or the data from some computer script. And so this is a very, very important issue with dealing with large-scale data sets. You can't put them into a paper, but you need to connect the paper to them, which is an issue that I think is very important. So I'll just say I'm going to talk a lot about ENCODE today, and this is just a big project. It sort of carried on from the Human Genome Project that finished about a decade ago, and this was the covers of Science and Nature, where they published the genome sequence. And the project essentially had two pilots to it. One was on 1% of the human genome, where they analyzed it with a lot of functional genomics, and we're going to talk about that. And another part of it was looking at simple model organisms like worms and flies. And those two pilots kind of came together into this sort of overall annotation of the genome that came out last year. So let me tell you a little bit about the structuring of the data, and I'm going to bring up this idea of what I call low-level and high-level data. And I'm going to come back to this later, and maybe you've all seen this, but I just want to make sure that people get this, because I think the structuring of the data, before you can mine data and apply statistics to data, you really have to think about what it looks like. And I think that there's a kind of natural progression to it that's just worth talking about. So you probably all know that the actual data that we get it looks like this, okay? What is this? This is something from a FASTQ file where you get reads on the genome, and then there's some little bit of, the quality of the sequencer, how accurate that base is, and so on and so forth. And so people take these reads, and they take the reads and they map them onto the genome, and then they interpret that mapping. So the mapping could represent the amount of binding, it could represent the amount of expression if you're in a functional genomics context. If you're doing a resequencing of the genome, you're just looking at the mapping, and then you're looking for the difference of a base at a particular point to find a variant. But so the first thing they do is map them onto the genome. And then if you're looking for sort of an enrichment, you'll get if you can sum up the kind of depth of your mapping at any point, you'll get a kind of signal, and that's usually called a kind of signal track. And so now you can see we're simplifying fairly radically our original data. Now then, you get all these signal tracks, right, for the, say, different factors and how they bind to the genome or, and so forth. And then the next thing people do is there's different higher level simplifications. So one thing people might do is they might aggregate these factors. They might average them around anchor points, and this creates aggregation plots. Another thing they might do is they might look at the factor bindings and try to put these into the framework of networks. And we're going to be talking about some of these simplifications. But I think it's important to realize these are kind of tertiary simplifications, or simplifications on top of simplifications. Okay? Within the framework of an RNA-seq experiment, you have the same type of thing. Here's the signal on the genome. But people often just summarize this whole signal on the genome. They'll kind of average it, average the amount of expression over an exon or over a gene into an RPKM value and so forth. Again, a big simplification. And I have a kind of way I like to think about this kind of genome annotation processes. You sort of start with the reads, you get that noisy signal, you process the signal. And then from the processed signal, people often, they find little blocks on the genome, okay, just sort of regions where the signal is high, and they might aggregate these blocks together and so forth. And this is, a lot of what you see happening in a lot of genome annotation processes. Okay, so that was a very verbose beginning that was just trying to say, well, I guess this class is about statistical methods in functional genomics, and I wanted to put it into this perspective of big data and kind of how you think about, we're trying to get to the structure of the big data. And so what I'm going to do now is I'm going to give you kind of three illustrations of practically how we use these statistical methods to grapple with the big data in functional genomics. And so first what I'm going to talk about is kind of building statistical models. And I'm going to talk about building a model that relates the input on the genome, the binding of a factor, to the output, the RNA-seq. So I'm going to talk about building statistical models here. Okay? And then the second part, I'm going to talk about trying to think about a structure that we can put on the data, and that's this network structure, and how that structure really helps us interpret the data. And so I'm going to talk about how we build networks from the data, statistically. I'm going to talk about how we can use those networks to interpret them and get actual useful things. And then I'm going to talk a lot about how we can see the impact of variation and constraint on the network. And then the final part of the talk, first I'm just going to do a little bit of a run through of some very practical tools that people have developed for processing this data. Just kind of a quick run through for chip-seq and RNA-seq data. And then what I'm going to do is I want to drill into two aspects. One thing is kind of privacy aspects, which is an interesting aspect of this type of data. And then I want to talk about allelic effects, which is a very specialized technical way of treating the data, which brings a lot of things together. So those are the things that hopefully I'll talk about, and hopefully you won't go to sleep too quickly on. Okay, so first of all, let's talk about the statistical models. Now this is really interesting because this slide is completely corrupted on my screen, but it came out perfectly on this screen, which is fine. I don't know why it's corrupted, so I will not look at the screen in front of me. So I'm going to talk to you about sort of a tertiary or even quaternary type of representation of the underlying data. So you have I'm imagining that you guys are learning how to think about the reads, how to process them into signals, how to call peaks and things like that. And now I want to talk about, well, once you get all of the inputs on the genome, and you also process your RNA-seq experiments and you get the outputs, how do you interrelate them? So what we've worked a lot on is building statistical models that interrelate them. So I'm going to talk to you about two types of statistical models. First, let's focus on a statistical model that relates the chip-seq inputs from, say, a histone mark experiment to the outputs. Now, the histone mark, the histone mark experiment gives you a sense of the degree to openness or closedness of the chromatin around a gene, and that is very related to the degree to which that gene is going to be transcribed. And so what you can do is you can go to the beginning of a gene, like the start site of a gene, And you can chunk it up into little bins around that thing, and you can just tabulate how much you have of a particular histone mark from a chip-seq experiment. Okay? And from looking at that, you can kind of take that as a kind of feature to kind of predict the level of gene expression. Maybe if you have a lot of a particular feature like H3K4, ME2, maybe you'll have a lot of expression of the gene. And you can do that for a number of different genes to try to predict the gene expression level. Okay? Now, you can do this individually for one gene, but you can also do it in aggregate, kind of averaged in a statistical way for many, many genes. And so if we talk about it, let's just talk about what the data looks like in an average fashion. So remember I talked to you about these aggregation plots. So imagine we took the TSS of every gene, and we averaged the level of, say, this particular mark, H2K4 ME2, around that gene, around the TSS. Okay? And this picture shows that value average for the highly expressed genes and the lowly expressed genes. And it's obvious from looking at the picture that the highly expressed genes tend to have lots of this mark upstream and the lowly expressed ones don't. Okay? So then what you can do is you can take all the different marks that people look at. These are many of the, there's this kind of zoo of histone marks. And you can take all the marks and you can go to each of these bins and put into the bin the correlation of the mark with the level of gene expression averaged over all the genes. And then you'll get a picture like this. And what this picture will show is it shows that, for instance, at a particular bin here, the level of that mark is highly correlated with gene expression, and that correlation falls off as you go away from the TSS. And for, say, this mark over here, it's actually anti-correlated. And so you can see there's a kind of positional effect, and you can see that some marks are correlated and other marks are anti-correlated. And then what people did with this is they were able to then put all of these things together, to put all the correlations together into a model. Now, I should probably tell you a little bit more about the structural model. I don't really go into the machinery of it, but you could imagine just building a simple linear model here, right? You could take the level of correlation in each of these bins, and you could just kind of add them up, right? You could add up all these things that are correlated, you could build a linear model. Right? Now, if you wanted to be cooler, or if you wanted to do something beyond a linear model, you could build a nonlinear model, which is basically just adding up all these things into a model, but you're going to have nonlinear terms. And so one way of building a nonlinear model is to build one popularly used one is a thing called support vector regression, where you're doing regression, which is building like a linear model, but now you're doing it in this kind of with this kernel, so that it becomes nonlinear. So and then what you can do is after you build your model, you can predict the level of expression of the gene, and then you can compare the level of expression to what you actually observe. And so this picture actually shows for all the genes in the human genome, this shows the predicted versus observed level. And so each dot represents a gene, and you just have how much you measure, how much you predict. And this picture was published in the ENCODE rollout. And the amazing thing about this picture that's really cool is this number over here, okay? It gets actually a pretty good, these are pretty good statistics for how predictable things are. I mean, in the framework of how we can predict things in biology, this, Pearson's R of 0.9 is very good. So that says that, we can actually account for a lot of the variation in the gene expression levels just from looking at the upstream marks. And then what we can do is, since we have a model, we can ask in the model how much are each of the terms in the model weighted, and those terms are associated with different histone marks, and this shows the weighting in the model for the different marks. And the weightings is useful because it says which of the marks is most related to gene expression. And so you see here, for instance, this mark, H3K36 ME3, is fairly important or something like that. Whereas this mark over here, H3K4 ME1, which you might recognize as a well-known enhancer mark, which is a little bit far away from the gene, is not so important. And then the next thing you can do is
What's the meaning of aggregation?
MS, a mean square error, root mean square error. Yeah, yeah. So these are different. Yeah, that's right. So these are different different statistics that show how well that line fits through the data cloud. So people have different statistics for this. So one of the most common statistics, which is the one that most people are accustomed to, is just the Pearson's R value. Okay, but there are other statistics you can pick, and actually this slide lists a lot of things. It's not completely clear what this is, but one of the statistics is kind of like if you kind of calculated the squared error off all these points from that line, what you'd get, and that's what that statistic shows in some normalized fashion. It's not as interpretable, I think, in a simple way. So actually what I'll do, to help you with questions, what I'll do is, because you're about to ask a question, what I'll do is, if you let me get to this next black slide, but I'll finish I'll stop at each, the way I've laid out the talk is, I constantly come to these little black slides where I come to the section boundaries, and what I'll do is I'll stop at each section boundary, and then we can go over questions for that little section so that you get a kind of coherent picture of the section. So just real quick, the basic idea I described. Okay, so then there's just games you can play, and I'll just quickly say some of the games. The games are kind of fun. You can do things like, I mean, once you build a statistical model that really relates things, you can play with the model. You can look at which histone marks are important in the model. You can also look at which bins, which areas around the TSS are the most important for the prediction. And it's actually very interesting. You see right at the TSS, that bin is the most important, which kind of makes a lot of sense. You can also do things like, you can build in addition to regression models, you can do classification models, where you just predict is it highly expressed, lowly expressed, okay? And then in those types of models, you usually use a different statistic, something called an AUC value, to talk about how successful they are. And one of the really neat things about this model was, I think one of the neatest things about it was that you could take the model that you tested and trained on protein coding genes, and you could take the parameters that you learned on protein coding genes, and then just go over to non-coding RNAs, and those parameters would, without changing them, without touching them, they would actually work fairly well to predict non-coding RNA expression. So this is not as good of an R, but it's still, there's a substantial correlation here. This is for predicting non-coding RNA expression. And then another game that you can play, which is kind of fun, is you could take, so the way I've described it is with the histone marks, we have the gene expression. They're in a particular cell. They're in a particular type of cell. But another game that you could play is you could say, I'll take the epigenetics on one cell, and I'll try to predict the gene expression on another cell. I mean, it's kind of stupid in a way, but it doesn't work as well as if you take a match thing, and that's what this picture shows here. So the blue line is the match, and then the other lines are not matched, and that's kind of satisfying. And then you can do this type of thing in different organisms, and you can, do it in mouse, you can do it in flies, and so on and so forth. So that's so that was the overall picture of this little histone model thing. So any questions on that?
It's pretty amazing the correlation between different histones and the gene expression. I was just curious that if it's always not word histone marks are uniquely not word genes only have one histone mark. Do you try to classify each of them and if there's any how many levels of histone marks are actually there, or how many groups of histone marks?
Well, that's a very interesting point. You mean maybe there's sort of like some histone marks that go together or something like that. And that's certainly interesting. I mean, there's we did that a little bit in our research, and there's definitely a world of people that study the chromatin stuff that are very interested in like histone code is sometimes the word they use. Now, the mindset of the histone code, which we did not find that useful, but is worthless. The way we set things up is we took each mark, and just think of a linear model, just add them together. So there's no cross terms. There's no term that says mark H3K4Me2 multiplied by H3K4Me3. If there were a cross term, that would imply, and if such a cross term were important, it would imply that there's a significant interaction between those two things. Now, it turns out, I didn't show you these in the slides, but I just mentioned, when we built these models, we actually looked for those cross terms. We looked at all the cross terms. And none of the cross terms actually had substantial loading. They had substantial weight. And the implication of that is fairly important biologically. The implication was that there isn't that much kind of interaction between these two marks. And that's, I think, a very powerful way that you can use the models to kind of get at your biology. Any other questions?
How did you divide the data to first learn and then validate the classifier?
Oh, this is more of an application thing, so I've kind of glossed over a lot of the methodological aspects. But yeah, a big part of this is what you just alluded to. So to do this right, you have to do what's called cross-validated training and testing. And so what usually, and now the good news here is that there's so much data, it's really easy to do this. And so you can literally, you don't have to do anything that complicated. You literally have 20,000 genes. You can build your model on 10,000 genes. I mean, 10,000 is plenty. And then you test it on the other 10,000. You know, or you can do more, there's more sophisticated things like, you know, jackknives and bootstraps and all sorts of things where you leave out different sections of the data. But the basic idea is you train the model on a set of the data, you test it on another set of the data. Any other? Okay, so now what I'm going to do now is I'm going to tell you about the same thing. You can do the same thing for transcription factors as you do for histone marks. So here's the same picture. This is for a bunch of, this picture actually is not from the human genome, it's from the worm genome, but it's the basic same idea. As you can look around the transcription start site, you can look at little bins, you can look at the binding of the overall binding of transcription factors, and you can correlate them with the level of gene expression. And it's actually, this is quite notable, and you see how it's very, very peaked around the TSS. This is the promoter, right? And you can see, except for these two factors, CEH14 and ALR1, most of the things really bind or, you know, kind of hug the promoter, right? And then you can see, which is also kind of nice, that some of these are very clearly blue and some of them are clearly red, and that implies that some of the transcription factors are very easy to see as activators versus repressors. So obviously a repressor, it's going to be blue. The more it binds, the less gene expression. Activator is going to be red. So this is just a nice way of getting at that is something. Now, for a given individual gene, something, even though overall a factor might be an activator, for one gene it could be a repressor in that context. But overall, this thing is kind of classifying things into activators or repressors. And then you build a little model. This particular model for transcription factors has to be a little bit more complicated model. So what it does here is it takes each of the bins, it takes the transcription factors binding in that bin, which is many transcription factors. It tries to predict the expression, and then it kind of has like an ensembling step, or kind of it takes the prediction for each bin, right, and then puts those predictions together into another classifier to make a kind of combined prediction. So this is a very common type of technique where people will put together a number of layers of predictors together. And it seems very baroque and kind of complicated, but the unfortunate truth is what a lot of these machine learning competitions have shown is that this type of baroque structure increases your prediction performance. So there's a very famous challenge called the Netflix challenge, and one of the things people learned from this challenge was that many, many layers of ensembling tended to do very well, which is kind of not intuitive, but the fact. And so this picture just shows the results on the human genome, and you can see here's your R value, there's the RMSE value. But the key thing is R value is still good, but it's a little bit lower. And then you have the weightings on the different TFs. And you can see some of the TFs are stronger in the model, others are not so much. But what's actually interesting, I should say, about this picture, which is not obvious, but I have to say a little more, and it's kind of neat, is this is a picture that is based on, this particular picture is based on the encode rollout data. And this was in the encode paper, and it's in this particular cell called K562, which is a kind of cancerous cell. And in this cell, I think encode must have studied something like maybe 70 or 80 transcription factors. So, and actually, this makes it even a little bit less, but I think that most of them are listed here. And the neat thing is that you can predict gene expression so well with so few transcription factors. And you might say, well, what do you mean? Well, there's about 1,000, maybe 1,200 transcription factors in the human genome, or maybe even more. And you might think you need every single one of those transcription factors to figure out the gene expression level, right? But here, you're actually getting a very good prediction of gene expression level by only using maybe about a 10th, or less than a 10th, maybe a 20th of the factors. And that's kind of not intuitive, why such a small number of factors can actually give rise to such a good prediction. And I have this is more of a very research thing. I have my own personal rationales why this works so well. It's not at all obvious. So I have one or two more slides, and then I'll go to questions if anyone has that. I just say you can also predict differential expression in addition to predicting the expression. So if you have two cell lines, you can look at the change in the level of binding in a bin and try to use that to predict the change in the level of expression. Same type of thing. And that's a little bit more mechanistic, or a little bit more active, because you're saying this transcription factor is changing, and that change is related to the gene expression level, and that actually does fairly well when you do that. And actually, again, one of the key lessons that you should sort of get from this is that if you build a model, the important thing, I mean, I'm not saying all the statistical modeling is easy, but in a sense, you know, you run these things, you can run them in R, you can understand it, but the important thing is to actually think about them biologically. And the nice thing about these models is if you really think about them biologically, they tell you a lot, or you can kind of understand them. And so this picture says, well, if I try to predict the change in gene expression, which factors are actually important for predicting the change in gene expression? That's the green things, right? And I'm predicting the change in gene expression from these two cells, which are the blue cell and the red cell. It's actually K562 and GM12878. And what's neat is all the things that get a very high value for the green, right, have a fairly high value for the red and the blue. So the implication is if a factor is not important in one of the cells, it can't be useful for the differential gene expression prediction, which kind of makes sense. And you can actually then look at, then you can correlate this with the expression of the factors, and you can see if the factor is expressed in both cell types, it's the most valuable in terms of predicting the expression, or the change in expression, than a factor that's not expressed in the cell. Then you can, of course, compare the histone model and the TF model that I talked about. And so it's actually, again, just very interesting biologically to do this. So, for instance, here's the profile of how important each bin is for the TF model, this blue line. And you see it peaks very much the TSS. So those little bins right around the TSS are the biggest contributors to the model. That's where, on average, the control box for the gene lives. That's where the most control over the gene expression of the gene lives, right here at the TSS, where you'd expect. What's interesting, though, is how different the histone model looks. Now, the histone model also peaks at the TSS, but it doesn't peak nearly as steeply. And you see the histone markings all over the gene, and actually even after the TTS are actually still relevant for predicting the gene expression. And so really, just the contrast, I think, is nice to get a sense of really the difference between the TFs and the histone marks. And then the other thing that's kind of neat is you might think to yourself, I've described two models, this transcription factor model and this histone model. Maybe I could put them together. But what's interesting is if I put them together, if I combine them, I get this combined model that has a prediction efficiency shown by the black line. And notice how the black line doesn't go any higher than either the blue line or the green line. And the implication is that I don't actually do better by combining the models than I do by keeping them separate. And that implies that there's a lot of redundancy between the two models. So it's not obvious, like when you sort of look at them, it's not completely obvious, but in a statistical way, the models are actually fairly redundant. So you don't get any more information by putting them together, which is a nice thing to see. And again, it's one of these things that you only really see if you do it in a proper statistical way. So just to summarize this section, I wanted to show you how you can build these, what I call tertiary or kind of quaternary type of annotations, these models that sit on top of all the processing above that provide some interpretation of how the inputs and the outputs go together. And I've talked about how you can do them for histomodels, you can do it for transcription vector binding, you can apply them in many contexts. You can take the protein coding model and actually use it for the RNAs. You can take the model without tweaking the parameters. And the model, the structure of the model itself tells you a lot about, you can study the structure of the model itself. It tells you about which histomarks and TFs are important, which histomarks tend or tend not to go together well, how redundant the transcription factors and the histomarks are between themselves. And there's this interesting result that actually a fairly small number of transcription factors are actually surprisingly predictive, which isn't immediately intuitive, and maybe I'll describe that in a little more detail later. Okay, so that was the first part, and I don't know how long we want to go on this, but should I keep going, or?
Yeah, well, for another 20 minutes or so.
Yeah, I'll go for a little bit, and then so this, don't worry, we're not going to do this all right now. So the idea is that at some point in this outline, we'll just kind of have dinner, and, we'll relax, and so what would be good to do, I think, is just to find a suitable ending spot, either maybe somewhere in the middle here, or maybe we get here, probably not, maybe somewhere in the middle there. We'll just have a break point and stuff like that. So now what I'm going to do is I'm going to shift gears. And so I talked to you first about models and how we can use statistical models to interrelate these types of data. And now I'm going to shift gears, and I'm going to talk about just a different concept of how we can put a lot of this stuff together. And this is the networks concept. So the models concept is very much based on the interrelating different tracks. It's very much like elements. We have the binding sites, we have sort of levels of expression here. They're expressed in kind of the linear track view. And this is the UCSC genome browser that kind of shows these tracks. And one thing people think a lot about in genomics is they think, is this track type of thing, does it scale? If we keep doing experiments, we do more and more experiments. We do 1,000 more transcription factor chip-seq experiments, and we do many different cell lines, and then we do more RNA-seq experiments. We're just going to essentially pile up more tracks, right? And then we start to wonder about, is that useful? Now, to some degree, we can encapsulate the tracks in these models, right? So the tracks themselves are, of course, simplifications, but then we can build a simplification on top of that, which are the models. But we might ask, is there other simplifications we can build? And so one simplification I'm going to talk about now is the simplification of networks. And so the networks idea is kind of, I like to think of it as a kind of a practical approach right now to interpreting data. So we have these 1D tracks, okay, that's where genomics is now. And, the real molecular biologist, what do they want to understand? They want to understand the molecules, right? And to really understand the molecules, these aren't molecules, right? This is not what molecules look like. Molecules look like this. And real understanding is, this is what the molecules look like. This is how polymerase is working. This is how the DNA is working as a big molecule. That's the goal. Now, of course, to do that on a very large scale, to do that for 20,000 genes in the genome, to do that for everything is an extremely ambitious thing. And you have to do it in many time points, in many cells. It's very, very hard. We're not going to be seeing this anytime soon. Okay? So the networks representation is kind of midway. It provides a little bit richer representation. It's kind of like a wiring diagram. I think it's like a 2D-ish wiring diagram. So it provides a little richer representation than the 1D stuff, but it's not so challenging as actually thinking about them in molecular terms. And, I like to tell people also that networks, I think, is a really good way to talk about biology because people use networks in many different contexts. They'll use networks to talk about the brain, to talk about computers, they'll talk about food webs, and of course, there are social networks and stuff like that. And so this picture of networks kind of transcends the particular discipline we're working in. And the fact that it transcends that discipline is very powerful because it means that we can communicate ideas to other disciplines, and we can also, as I'm going to spend a little while about, we can use analogies and comparisons in a very powerful way to give us intuition about the biological context. And so let me talk to you a little bit about some analogies, and I'm going to come back to them. So what we often do, what do we do in sort of regulatory network analysis? What we want to understand is we want to understand kind of like what's the causal regulator, what's the master regulator? How are things, what transcription factors driving everything? So how do we get intuition for how does that work in the system? Well, here's a system that I got from the New York Times, and this is from a few years ago, and it's called the blame game. And what it has is it has arrows that show blame, where one person is blaming another person. And the neat thing about this picture is maybe you realize what these people are blaming each other for, maybe you know who these people are. But the really nice thing about this picture is just by kind of getting the concept of blame and looking at the structure of the arrows, you immediately kind of understand who's in charge. You know what I'm saying? Who's the master regulator in this figure? And the thing is people immediately get that. You know what I'm saying? They kind of get that. And that intuition is very powerful if you can then apply it now into a biological context. And here's another one. This is a picture that also is a sort of surfaced around the same time in the New York Times. This one is actually guilt by association. So I don't know if you know who this guy is. He's someone who got in trouble, and so. But the point is, and then what they did, and this is literally what they did, they made a picture of all of his connections, and then they start to look at guilt by association. That's kind of what we do, right, in the sort of clustering analysis. So I'm going to come back to those analogies. So now what I want to do is first tell you how we're going to build a network from the tracks. Okay? And then what I'm going to do is study the network and then talk a little bit about analogies. So first of all, how do we build the network? Well, remember, I'm of course going to gloss over the actual experimental work done, which is seriously hard work and seriously important. And I'm just going to say that in the group of people I was working with, we were given this very large data set, which consists of about 120 transcription factors in about 460 experiments. Now, this is a huge data set. It's definitely a very big amount of data to pour through. And so the data, what does it look like? Well, it has these signal tracks, and then you can find regions in the signal tracks that have peaks. And I'll talk to you a little bit about how you can find peaks later, but just the simple level is you can find about 7 million peaks in those tracks. And so one thing you can do is you can just make up for, say, one particular factor, this complicated map that looks like this, where you have all the factors that are binding, say, near where one factor binds. You can see some bind in a similar way to that factor and whatnot. And that's sort of a little bit more of a representation of the data. And one of the initial things people did is they tried to cluster the factors with each other. So they would look for the combinatorial association of transcription factors. So you could take all the transcription factors and you could say which factor tends to bind with which factor. And so this is a big heat map that shows these factors can come together, and here's some examples of factors that come together, and then there's some factors that come together in one cell type but not in another cell type, and so on and so forth. And so you could just simply correlate them. Now, it turns out, if, I'm not going to go into this in any detail, but if you simply correlating the binding of factors, it gives you kind of a co-occurrence matrix. But you can do a little bit better than that, and people can do more sophisticated things than necessarily correlating things. And so one of the things that was done in the encode context was kind of trying to, again, build a statistical model that would predict the co-binding map from one factor. And in the statistical model, you could have the relative importance of all the other factors in that model. And that relative importance gave you essentially a slightly more sophisticated co-binding score, and then you could cluster that co-binding score. I won't go into this in detail, but I just wanted to give you the idea that you can do a little bit more than that. So one thing you can do is you can look at co-binding, and that will give you kind of co-association matrices. Now another type of association you can look at, which is probably the most direct, is you can try to look at the association between a factor and the targets it regulates. And so some of the regulations are very simple to understand. Those would have to do with, say, a factor that's binding right upstream of a target. And just like we talked about for those models of gene expression. And so we would call this type of binding a kind of proximal binding, okay? And so if you look at all the proximally located sites and you construct all the bindings from transcription factors through the proximal site to the target gene, you'll get a massive, massive hairball. In this particular context, where we start off with about 100 and some odd factors, we'll end up with a hairball that consists of about 500,000 edges. So there's 500,000 factor to target regulations here. Okay, so a big hairball. And so the first thing we might want to do is prune that hairball into stronger proximal edges. And I'm going to describe a little bit about how we can prune that statistically to go from 500,000 edges to maybe 25,000 edges. So that's one thing we can do. Another thing that comes up is, what do we do with a peak or a binding site that's far away from a gene? Okay, what can we do with that? Well, that binding site could be located in a region that people call a distal enhancer, a place that's far away from a gene, but maybe it loops, the DNA loops in a certain way, or somehow it exerts a kind of action far away on the level of expression of the gene. And so we can also try to find linkages, you might call them distal edges, from an enhancer binding site to the gene, and then from the transcription factor to the enhancer binding site. So I'm going to talk a little bit about how we do these two operations. And this is a little bit technical, just to give you a little bit of a flavor of how this is done. So, okay, so now we want to find, first let's talk about how we find the strong proximal edges. So one of the things we might do is, the simplest thing we might do is just look at the strength of the binding signal right upstream of the transcription factor. In fact, we don't even have to look at the peaks. We can just take the total amount of binding signal, just the amount of binding of a factor upstream of the thing, and just add it up. That would be one simple measure of the total amount of strength. And then we could threshold that total amount of binding in a particular way. But one issue that's a little tricky is that some transcription factors tend to bind right near the TSS, and other factors, they're a little bit more, this isn't so much an enhancer thing, but they tend to have a more distributed binding. So one refinement to this is we can make up an average profile for how each factor tends to bind, and then we can multiply the binding signal at a given gene with the average way the factor tends to bind, and then we can take this kind of weighted average and use that to get an overall score for binding. And then we can threshold this score, somehow look at the distribution of these scores and threshold to get a kind of overall regulatory score. Okay, so that's how we might find the strongest binding. Then now this is a very techish and complicated slide, and I'm not, if you have a test, you can be tested on this. This will drive you crazy. It's just too confusing. But I will try to describe this slide in simple words. And so this slide talks about how we can identify distal edges. So to identify distal edges, we first have to find enhancers. Okay, enhancers are places that are far away from genes where there's lots of binding. So if we had all the transcription factors, we would just find regions where there's lots of binding, kind of clumped away from gene. Okay, that would be the simple thing to do. But we don't have all the transcription factors. So what do? What we do is a little bit more complicated. We take the binding that we observe, the clumped binding where we observe distal for genes, from the, say, 100 transcription factors. We use that as training data. So these are potential enhancers, okay, but they're not all the enhancers. So use that as training data to train a model that looks at the chromatin structure around those things. And so now we'll learn the types of chromatin features that tend to occur around those clumped binding regions distal from genes. Now we take that chromatin model and we go out and we look for other regions, we call these binding active regions, that are away from genes. Okay, so these regions might or might not have a lot of transcription factor binding, but they have the chromatin characteristic of enhancers. And then what we do is we look if there's at least one transcription factor binding in that region. Okay, so that's now we have a putative enhancer. We have a transcription factor binding to that enhancer. Okay, but how do we know how the enhancer connects to the gene? Then the next trick we do is we say we look at over a whole bunch of different conditions or different cell lines. We look at how the chromatin in that enhancer is going up and down relative to the gene expression level of nearby genes, and that links that enhancer to a gene. Okay, so now so the overall idea is we find about 130,000 enhancers from the chromatin structure, and then we can link about 20,000 of these enhancers to genes, and then if a transcription factor binds an enhancer, we can then link that transcription factor through the enhancer to the gene to create what was called a distal edge. Okay, I know that was a little confusing. So now we get these networks of about 20,000 distal edges and 25,000 proximal edges, and now we're going to analyze these with kind of network theory, whatever that means. And so let me tell you a little about network theory. So there's some ideas here that are very simple. One of the ideas that you've probably all heard of is the concept of the hub. The hub is the thing that has lots of other things connected to it. So for instance, this green thing here is a hub. There's a lot of connectivity. It has what's called a high degree. This thing is also a hub. But another very important concept, which is not as intuitive, is the concept of bottleneck. And a bottleneck is a place in a network where most of the shortest paths in the network go through. So the number of shortest paths in the network going through a given point is called the betweenness of that point. And a place with maximum betweenness is called a bottleneck. Now, how do you get some intuition for that? Let's talk about this structure here. Now, I don't know if people realize what this structure is. Maybe Harmon knows what this is. That's the George Washington Bridge. So this is the George Washington Bridge. So this structure is a bottleneck. And why is the structure a bottleneck? The structure is a bottleneck because if you visualize all the trips going up and down the East Coast of the United States, okay? Think about every possible car trip, from your house to house in Washington, or this house, all over the place. Think about all those possible trips, and think about places that all those trips have to go through that have maximum betweenness. Well, there's not that many places you can cross the Hudson River. It is just not that many. I think it's like 7 or something, right? And this is the biggest one, right? So most of those trips through, up and down the East Coast are going to go through that structure, right? And therefore, you get information flow bottleneck. Okay? And so this is important concept in thinking about transcription factors because transcription factors are all about information flow. And so this concept you'll see is very useful. So what's another concept that's very important? Transcription factors create what's called a directed network. So you probably so in the previous picture, we have what's called an undirected network. Things are just touching, right? But transcription factors create what's called a directed network because in this context, one thing tells another one what to do. So there's an arrow, A tells B what to do. And we're very used to, in a social context, this kind of org chart mindset of like, who's my boss? And this is mostly in the corporate America mindset. So, you've probably all seen an org chart. Where do you sit on the org chart? How many people do you report to, and so forth. And so here's an org chart. This is the old computer land org chart of Apple Computer. And Steve Jobs is in the middle of the org chart here. You see he's the kind of Death Star, and he regulates all these people. And I think Tim Cook is somewhere here. I don't know if it's on this thing. He was promoted into the center. But in any case, you're very used to this picture. So what we wanted to do is build an org chart-like picture for transcription factors because this is what we're very used to, a way we're used to socially thinking about directed networks. So how do we do that? Well, one thing we can do is for each transcription factor, remember we're just given a kind of hairball to start with. So we're given a picture that looks like this. So how do we turn this into a hierarchy? And so one thing we might do is for each transcription factor, we might say, how many things do you regulate versus how many things regulate you? And let's just stay within the realm of regulators or transcription factors. So we're going to say, for each transcription factor, how many things regulate it? its in degree from other transcription factors, and how many other factors, just transcription factors, does it regulate? That's its out degree. And we can build a little statistic that talks about, you know, how much larger the out degree is relative to the in degree. Okay, so I think what I'm going to do, if I can get to one more subsection, I think that would be good. So any questions on this last subsection where we just talked about motivating, constructing a regulatory node?
Quick question. When you
Sure.
mentioned that you find only like about 10 transcription factors kind of predicting gene expression, are those factors like similar across cell lines? Because it seems to remind me that no matter the unknown functions of these transcription factors, conserving those general transcription factors which override the work in other tissue-specific.
That's a good question. I mean, the question is, are the small number of transcription factors that predict gene expression, are they more, less cell line specific? Is that what you're, and that's an interesting question. I don't know if we have the complete data set that we could use for that, but interestingly, that I don't think is the case or something like that. So here's the reason it's not the case. It's not that there's a small group of transcription factors that will predict gene expression. The really, I didn't describe this, but the result, which is non-intuitive, is any small group of transcription factors. So if you can take any group of 5 or 10 transcription factors, any group, and that group will actually be fairly successful predicting. This is not intuitive at all. It's not obvious at all why this should work, but it does. And I have actually, if people want, I can give you my biological rationale for that. It's not obvious, but it's like I say, it's completely not obvious why that should be.
But you think that, you know, chromatin accessibility would correlate with the chip signal as well as expression, right? So to share, is that the kind of direction we should?
Well, no. Well, so my rationale is that the chromatin, it makes sense. So the chromatin basically says the openness of the upstream region of the gene is related to how much is expressed. That certainly makes sense. But why is it that just 5 transcription factors can predict gene expression? Well, how about, imagine this scenario. Imagine a scenario where you don't think of transcription factor binding as a static process, you think of it as a dynamic process. You think that what happens is there's a few key factors or combinations of factors that kind of pry open the DNA, and they open up the chromatin, which is all tightly packed, and they open it up, and they get it ready so that the polymerase can transcribe it. Once the chromatin is opened up, a whole bunch of other stuff just kind of opportunistically binds there, just binds there, okay? Now, a lot of stuff binds there, and so the point is, when we're doing the correlation, and we're seeing after the DNA has been pried open, so we're looking at all those things that are just, they tend to be binding upstream, but they're not the causal factors. They need the chromatin to be opened up, and so the specificity of the regulation is in more of this dynamic process where something opens up the chromatin, and that's not what we're seeing. We're seeing after the chromatin's been opened, a lot of factors tend to glom onto the open DNA. And that's why you can basically grab any collection, or I mean, in any, say, subset of 10, you're going to find a bunch that are just going to be, I wouldn't say they bind non-specifically, that's probably not a fair way to put it, but they're just binding to the open chromatin in that particular cell type. So that's my rationale. I don't know if it's true, but that's my rationale. Any other questions?
In your first slide, you had 2D, 3D, and 4D. By 4D, I'm assuming you mean MD simulation type of things?
That's correct. I meant with time. Like looking at a structure, and not only having the structure, but actually how it changes with time.
Confirmation. So is there any established or cool way of combining 2D, 3D, 4D information at this point?
Oh, well, there's certainly people who are this is much more in the structural biology world than obviously the genomics world. There's definitely communities of people that think a lot about, how the transcription factor really recognizes the DNA, how how the DNA structure changes, in the recognition process, how, this is changed by other things. It's very detailed, and on the major issue is that usually people who study this, they study one transcription factor for 20 years. I mean, quite literally. I mean, you can't do this on 20,000 things. So what happens is people get ideas and they get models from looking at one thing, and then they kind of, and this is of course the leap, they sort of say, well, that's the way everything works. And sometimes that's true, sometimes it's not true. And the unfortunate thing I think about a lot of structural biology is that a lot of these anecdotal pictures that are really well worked out, they actually don't have a lot of predictive generality. So like if you, you can study, I don't know, a particular zinc finger protein, ZNF279, your entire life. But you're not going to get from that one study a general thing that will let you predict where things are going to bind in the genome. It's just not going to work. Now, you get ideas, you know what I'm saying? And those, and the ideas, I know you just heard about the position weight matrix. They help you think a lot about maybe what's going on in that position weight matrix, but they're not going to provide, just in terms of like cross-validated prediction, they're not going to enable you to build any type of model that will have predictive success.
The thing is, the converse is also true, where by studying everything, you're not necessarily going to get at how each little thing works. So for instance, the data you're putting in here, some of these specific cell types, if you don't have, like a neuron, for instance, the transcription factor that turns on neural growth, that isn't even in your model.
No, that's correct. Well, that's completely correct. I mean, it's a little unfortunate, but the reality is that a lot of the large-scale models, which are very genetic, very correlative and stuff like that, they don't actually inform too much about the detailed mechanistic stuff going on. And likewise, unfortunately, the detailed mechanistic stuff doesn't inform as much as you'd like on the correlative structure. Now, the belief, of course, is that if you had total knowledge about any of these processes, they would connect, you know what I'm saying? But where we are at the moment is they're just kind of two perspectives. It's the landscape view or the microscope view. And, maybe 10 years from now, it'll be different, but that's where we are. And it's a little unfortunate because there's definitely some people that prefer, there's definitely a bunch of microscope people out there. That's certainly true. And they don't like the large scale stuff. But the reality is, if you want, you can't do anything with the small scale stuff right now in terms of building a type of predictor model or something. It just doesn't do anything. So.
In parallel to that, though, in order to construct any of these networks, you need to have something that tells you about what makes two things go near each other, right? And so to inform that, you need something that might have came empirically or theoretically, right? And another concern, I think, is that sometimes with the empirical data of how things come together may or may not have anything to do with biology underneath itself, right? Like two things that bind in a these two hybrid system may or may not bind in my immune cell at all. They may be totally unrelated, but in the network they're going to come up as really close to each other because of these two hybrids that affect the information.
That's correct. That's correct. Now, and just to be precise, in this context, the network we're talking about here, these aren't networks from the Easter hybrid, they're networks from the chip-seq, but it's the same idea. And one of the big criticisms people have, and I should have made this, I'll go back because I'll make it a little bit clearer. One of the big criticisms they have of the chip-seq is they'll say, why did I go from this picture of 500,000 to 25,000? So a lot of people look at the chip-seq and they say, oh, there's just a little bit of binding there. Do we really know that means that transcription factor is mechanistically really binding in front of that gene and turning it on, right? And so there's sort of a conservative bunch of people would say, let's just look at where the binding is really strong. There's a really big chip-seq signal there, and those are the ones that we are really going to use to build the network. So the picture here was we want to look at those chip-seq peaks that are really, really big and really strong and represent a lot of. It's not so much a statistical thing because even the little bumps are statistically supported over background. That's not the issue, but it's that there's this idea, which I think is true, is that a lot of even if there's statistical support for that, it doesn't represent kind of mechanistically important binding, what I'm saying, which I think is, a lot of people would say, I have a transcription factor, does it really bind, in front of 5,000 genes. You know, maybe it's really only binding strongly in front of 500 of them. So that was the mindset of going from the 500,000 to the 25,000, to really look at the strongest, to build the network just in the strongest binding. But that's a whole debate people have.
But why can't we layer up that information with like structural information that you get, let's say, from TDB database or so?
Certainly you can. I mean, well, actually, what people often do, you could do the structural information, but the simple thing people would often do with this network, the next step they would do is they would say, I demand that each edge of the network has a motif. You know, it's like I'm not even looking at the motifs. I'm just looking at there's a strong chip signal. Then they'll also say, oh, does the chip signal does it have motif support? You know, but now you're getting very model-oriented, very mechanistic oriented. You're demanding that you know the motif and that you can find that canonical motif. But fair enough. You know, so people then whittle their picture down further. And they'll say, this is a signal. It's very strong. There's a motif. You know, and again, the mindset of this tip procedure was I get a sense for each TF where it tends to bind around the TSS. Is it binding in a characteristic place and things like that? And so they'll use a lot of these, you know, simple ideas, but reasonable ideas to kind of whittle down the forest of all the binding into what they would imagine to be the important interactions. But that's sort of the, I mean, one of the criticisms people have of these chip-seq experiments is, you know, if you do the experiment and you do the statistics right and you process the data, you're going to get tons and tons of binding events. And so people will say, is that binding real? And the truth is, no one's ever validated every single little bit of binding functionally. It just doesn't happen. So, you know, there's different camps in terms of what people do. Like I said, the camp I represent is that we believe chip-seq data, but we're fairly conservative, and so we're going to try to focus most of our analysis on the stronger bits of that data or something like that. Other people will say every last bit of every statistically supported peak is, you know, they're all equal, which isn't, you know, or some people will say, I believe nothing. Well, you finish, then he goes, yeah.
It's a question, but so far, if I've understood you correctly, you've shown us data that depart from static measurements. You showed something about comparing two cell types, but this was static measurements. Do you feel that some of this could be enhanced by having dynamic measurements of how the genome responds to a certain stimulus, for example?
Oh, definitely. No, definitely. Well, that was one of the things in the models I was kind of pushing a little bit, the differential expression type of thing. I mean, I think having, you know, anything related to change is usually a little bit stronger. So like, you know, I mean, you know, it just requires the data. You know, of course, you'd have the data in one cell type, the matched data in another cell type. If you can see that the change in binding of a factor is related to the change in binding, or, well, is associated with the change in binding of a gene, that's a little bit stronger. It's not perfect, you know, it's still correlative, but it's a little bit better. And so certainly people like that, you know, for sure.
Yeah, that's good. Similar question. No similar questions.
The philosophical question or remark is that I think one of the reasons to want to drill down to the mechanism, like the underlying sequence, is that, for instance, you're really asking what is your goal in building a predictive model, right? Because of course, one extreme is to measure pol 2 occupancy and predict expression, right? But you're excluding pol 2. But you do have histone modifications that are pretty close to the expression in terms of the whole causal chain of events, right? So it's easier to get a good R-squared from that kind of model than from a purely sequence-based model, for instance, right? And I guess one thing that you couldn't predict is that we should use empirical data as the predictors, right? Is, for instance, the impact of SNPs in non-coding DNA, right? So I guess that would be a reason for wanting to
yeah, no, that's exactly.
Why do we see the binding at these positions, right?
That's exactly. I mean, I'm going to get to that in a little bit. That's actually going to be here a little bit. Oops. It's one of the points of these models, the networks, and also the statistical models, is to. I mean, I'm not describing this the way exactly people think, but a lot of the mindset of thought is that we have human variation. We have variation and we want to interpret it. And so the way we interpret the variation is we have a model or an annotation on the genome that talks about what that bit of the genome is doing. And then with that understanding of that bit of the genome, we can go back and look at different variants and kind of say, I mean, in the extreme, we would have a model, and we're not quite there yet, but we'd have a model that would say these variants maybe have a big effect, these variants don't have a big effect, and so on and so forth. And that's kind of what people would like as far as, it's not necessarily mechanistic, but it's useful, right? So people want to have a situation where they can go to a personal genome, which has millions of SNPs, and they want to interpret which of the SNPs are going to have an effect, or they want to look at different organisms and stuff like they create a mutation here. They want to know, is that mutation, what's it going to do? And so that's the idea. I'm not describing the motivation that way in this, but that's sort of at the back of people's mind.
Okay, well, I think it's a great point to take a break, and we'll start again at 7 sharp. Thanks to Mark.
What I want to do is I'm going to talk to you now a little more about the network stuff, and then at the end I want to go into this data and tools for genomics. So if you remember, I hope to jog your memory, we were talking about how we build the network, and then we were talking about sort of calculations of the network. So, so now we just start to put it together and actually just what do you get from this type of thing. So if you remember, we had this concept of the hierarchy and the directed, we had the concept of the org chart and the directed network and what sits up higher in the org chart. We made this statistic for how high you're sitting up. And if you look at this statistic, just this is an empirical thing, if you look at it just for the transcription factors in the human genome, it makes a trimodal distribution. And so then what you can do is you can say, well, that suggests that there's 3 levels to a network. And so what you can do is you can just do literally a simulated annealing procedure, where what you try to do is you take the transcription factors and you put them on 3 levels and you move them. You can just think of, to start, you can just think of it just randomly moving them between levels. And at each step, what you're trying to ask is, I want to move them so that I maximize the amount of downward pointing stuff relative to upward pointing stuff, right? Now, if you had a random network, just completely random, the amount of upward and downward stuff at any point, there would be no direction to the network, right? It would just be, you never could find a ordering where you had mostly downward pointing stuff, right? So in any case, you do this this procedure on this network, the simulated annealing, and you end up with this. And the impressive thing about this picture is that these are the transcription factors in the human genome. The lines are edges where one factor regulates another factor, and the color of the lines is green if it goes down and red if it goes up. And so the point of this picture is that, sort of obvious, there's much more green than there is red. And so by and large, you can actually build a hierarchy to the factors where there really is a group of factors that mostly sits on the top that has downward kind of flow to the next level and so forth. So like I said, if you had a random network, it wouldn't show this, but it turns out that the factors in the human genome have this natural hierarchical structure to them. And one consequence of the hierarchical structure is you build up a middle layer, and you can kind of imagine, you just think of the George Washington Bridge picture that we talked about earlier. The middle level is obviously going to contain more of the places where you have more surest paths going through, and so it naturally builds up kind of information flow bottlenecks, and you can measure that. This is the bottleneck score between this score in the middle. And then the other thing that you can do, which is kind of fun, is you can this is the network based on the proximal regulation, the 25,000 proximal edges. But now if I leave the factors in the same locations, and then I rewire them based on their connectivity in the 20,000 distal connections, I get this picture. And you can see it looks radically different now. Suddenly, there's almost about the same amount of red and green, and it doesn't look the same. And so this shows that the distal connectivity that you get with the enhancers is a very different structure than the proximal connectivity that you get through the normal rotors. And then you can ask, does this kind of org chart make some sense in terms of intuitions? And again, this is the kind of social intuition. So one idea you might have is that the factors at the top maybe are more influential, because of more influential things at the top. And you can measure influence actually very nicely from, remember, that model we talked about being the statistical model. So each transcription factor, remember, has a weight in that model. You know what I'm saying? It has the degree at which it drives gene expression. And so you can ask, what's the average weight in the model, which is essentially the average correlation of the factor with gene expression. And then you can paint each of the factors by that. And you can see just by eye, if you roughly scan this, you'll see that there's darker color ones slightly at the top relative to the bottom. And so the factors at the top tend to drive gene expression a little bit more than the ones at the bottom. And then you can ask, oh, maybe, you know, this again based on social intuitions, you know, maybe somehow the people at the top are better connected. And you can measure that too in terms of the factor hierarchy, in terms of you can measure the connectivity in other networks, say the protein interaction network, the phosphorylation network. And lo and behold, what you see is the factors at the top tend to be better connected than those at the bottom. And I'm going to skip that one just real quick because I think I can just jump to this slide. The other thing that you can do is you can look at, so that was the global wiring of the network. The other thing you can do is you can look at the kind of more local structure of the network. You can look at, say, any three triplet transcription factors, and you can look at how it's wired. And so this particular wiring of these three factors has, for instance, STAT1 regulating rungs X1. It also regulates STAT3, and STAT3 regulates this. And this creates what's called a feed-forward loop, where A regulates B, A regulates C, and C regulates B too. And you can ask for all possible triplets and all possible wirings of those triplets, you know, how often do you see that kind of little circuit board element in your global wiring diagram? And one of the things that's kind of interesting in this picture is that there's a tremendous enrichment, and that's what the number shown here. Actually, I can just do this one here. The number shown here is you can ask you can ask how enriched they are, and there's a tremendous enrichment for these kind of feed-forward loops. And people have ideas about why you might see a lot of feed-forward loops in the transcription factor regulatory network, and some of those ideas have to do with that this motif is very associated with kind of noise buffering or certain signal characteristics. Okay, so that's just some ideas on, we built the network, and you know, the thing that I think is very satisfying is you have all these mathematical ideas to build the network, but then you build it, and it makes a lot of sense. You know, it just, it's sort of intuitively understandable what has happened in terms of other properties. Now, the next section, what we're going to do is we're going to look at how variation impacts on the network. And I'm going to go through this a little quick because it's a little bit of a digression, and I don't want to go too long. Actually, before I do that, I should say, is there any, was there any questions on that section? So I'm just going to do this a little quickly, and like I probably should describe the background of this, and then what I'll do is I'll give you a sense of the background, and then maybe just kind of go through the results kind of quickly.
I'm just curious as to, I mean, this is, you know, you started out with sort of, you're up here on the big data and sort of moving down in terms of describing it, and I'm wondering if there's any thought about how you put this kind of information together with, you know, what's been gathered over the years about sort of how these things work, and sort of, you know, like you described, sort of the microscope level, right? You know, so for example, there's these, you know, very crude tools like, you know, IPA, right? Like that people try to, but I'm wondering if there's been other thoughts, sort of more sophisticated ways of thinking of how to integrate all the stuff that's been done by experimental biologists with big data, with, to see how,
Well,
what can we learn by that comparison?
Well, I do think that's interesting. I mean, so we have this hierarchy here of transcription factors, and we've argued that the hierarchy makes sense from more of this kind of large-scale correlative distinction in terms of the great degree to which things drive gene expression or they're connected. But there's definitely a lot of people who have done small-scale experiments. They'll say over and over again, "Oh, I mean, everyone who studies a transcription factor says their transcription factor is a master control factor." I mean, it's like you can read like 100 papers. Every single person's factor is the master control factor. But there is this sense, which is right. What I think you're saying is that people, when they do more small-scale experiments, they are thinking in terms of this type of picture. This is like a controlling factor for a whole process. And we haven't done this comprehensively, but one of the things you can do is you can take those anecdotal stories and map it onto this type of hierarchy and see, well, are the factors that people are anecdotally calling control factors for a particular process, do they sit, at the top of this type of hierarchy, or how do they relate to this overall hierarchy? And certainly in some, anecdotal situations, or for particular ones, you can see that, you can find a paper. I probably should have an example where a person says this isn't a master control factor, and it does indeed sit near the top of the hierarchy. But I don't know if we've comprehensively done that for, a sort of literature search on that and kind of compared it with this type of structure. But I do think that's an interesting thing to do. So there's definitely there's a lot of papers published where people will say, I want to understand differentiation in the cell. I want to understand, this particular process. And I looked at the regulation associated with that, and these factors are involved, and this one is controlled.
What's your view of how one could go the other way around and try to use this information to design experiments?
Well, I think very much you could do. I mean, and actually that's where I'm going to talk a little bit about it in the impact of variation. But the mindset very much is that, if you know things that are potentially more controlling, and you have the ability to disrupt them, or you have the ability to modify them and so forth, that you'll expect to maybe see a larger effect. You'll expect to, or you might want to target them for some type of thing. And that's actually exactly the mindset of the interrelating with sequence variation. So I'm going to unfortunately go through this a little quickly. This is actually a big topic, but the sort of broad idea is we have this hierarchy of things that we've constructed essentially from the functional genomics data. And, we think it makes some sense. And now what we want to do is we want to interrelate it with this vast amount of data, particularly available for humans, on variation. Okay? And so there's now variational data, I'm sure you could have a whole Cold Spring Harbor course on variational data because there's a lot of ways of thinking about it and so forth. But just one simple way of thinking about data is if you have a transcription factor and you have the binding sites associated with the factor, are those sites more or less conserved than other factors? So some factors, they're more sensitive to mutations than other factors. Now, that's a very simple statement. And you might say it's trivial to measure, and in a certain sense, you can say, well, how many mutations do I have in a given factor? How many mutations do I have in a given binding site? It doesn't work quite like that. There's a lot of biases in the data, and you have to think about exactly what you're measuring. But there's many measures people have of the level of variation in the sensitivity to mutation. So, for instance, one of the well-known ones, if you have two organisms, is something called the Ka-Ks ratio, which is basically the amount of non-synonymous or apparently functionally effective changes relative to synonymous changes, which are apparently neutral. And so you can the second thing kind of normalizes the former. In relation to population genetic data, you can look at the amount of SNPs in something. You can also look at sort of what's called the change in the allele frequency spectra. the idea is there that you tend to have more rare variance when something is under so much stronger selection. And there's a whole variety of these different measures. Listen, unfortunately, I'm not going to go into them in any great detail with you now, but the long and short is you can for any given transcription factor or its binding site, you can look at these measures. And one of the things you find when you do this is you find for many, essentially for all of these measures, both within the human population and across the primates, there's a very clear trend that you see in the networks that the more connectivity something has, that means the more the hubbier it is. Either that means the more things a factor regulates, the more factors that something regulates, the more constraint it's under. That means the more sensitive it is to mutations. Okay? And then when you go to the factor hierarchy, this is sort of related to that, but you can find a very nice trend that says the factors at the top tend to be under greater constraint than the factors at the bottom. And so this particular picture is drawn where the factors are drawn according to using this thing called SNP density, which is sort of the just a very simple metric of the number of SNPs in something. And you can see there's just less SNPs in the things at the top. And so the factors at the top are more conservative. Yes, maybe some social intuitions you might have, maybe not. And then I have this other little section here, which I'm going to go through a little quickly. It's a little peripheral, but I think it's kind of cool. And it really talks about how we can use these analogies and to really understand the impact of variation on a network. So what I've told you is that the overall result we get is that more connectivity is associated with more constraint. We find that in the human regulatory network. Now, people have published gargantuan numbers of papers on networks and variation, and I summarize some of the papers here. And in all the papers, they find in many different networks, whether it's protein interaction networks, microRNA target regulation networks, whether they're looking at E. coli, yeast, human, wherever, they found this trend of more connectivity is associated with more constraint. So this has been found in many, many contexts. And so, and it makes a lot of, it sort of makes sense. You know, if you have something that there's a lot of connections to, it makes sense that it's going to be more difficult to mutate. And that's what happens in biology. And you're like, well, that's great. And so, but we wanted to understand that process. And so what we did was a little weird, but it's just kind of bizarre. So what we decided is we wanted to compare the biological system, which is which is a sort of naturally created thing, and which evolves naturally, with a system that is similar but evolves very differently. And so we actually, and this is a little digression, so we compared, we decided that we want, since the genome is like a computer operating system, or so like a biological operating system, we were going to compare it to like an operating system of a computer. And this is a little out there, but I'll just show it to you. So the computer operating system in your computer actually has a regulatory network. The network in a computer operating system is called the call graph structure of the operating system. It talks about which routine calls another routine. And it turns out that you can actually download this entire structure because there's lots of freely available software codes. Like you probably know the Linux operating system you can freely get. And then you can do a little hierarchy for that, just like we showed you. Now this particular comparison, I'm just going to focus, as opposed to looking at the human hierarchy, I'm going to have the E. coli hierarchy, but it's essentially the same thing. The quick thing to mention is that the computer operating system hierarchy, which is shown over here, looks very different. It looks kind of like inverted relative to the natural hierarchy. Okay, and you can ask yourself, why is this? And this picture sort of talks about how the hub structure is very different, but the picture, just the intuitive picture of this is very easy to understand. In a biological system, the hubs in the network, or the things that have the greatest connectivity, are master control regulators. Like, for instance, in E. coli, there's a gene called CRP, which just controls a lot of processes. It regulates a lot. In computers, the things that have the most connectivity are actually the slaves. they're not the controllers. The routines that have the most connectivity are like the print routine that prints to the screen, or the memory allocation routine. Anyone who's programmed a computer will realize why this is. When you program computers, the way they're built is they're built very modular. So there's these little building blocks that lots of different master control proteins constantly call. Okay, so it's a very inverted idea. So there's in computers, there's a few slaves that are controlled by many masters. In biology, there's many slaves controlled by a few masters. And then what's really, really neat is that if you take the Linux operating system, you can actually watch it evolve, right? Because you can download every single version of this operating system from the moment it was born, right? And you can just look at it and you can see how it changes. And so we developed a measure of mutation in this operating system. And so we looked at each routine and we looked at how much it changed, just like the way a gene mutates or a binding site mutates. Okay. So if you look at just the distribution of these measures, things start to be a little interesting. So like here's the distribution of what's called DNDS ratios for biological system, where most things are under what's called purifying selection. That means the functionally effective changes are actually much less than the neutral changes, and only a few things are evolving very quickly, where there's more functionally effective changes than neutrally evolving changes. Okay? In the computer operating system, it's similar but not exactly. So there's some routines, like for instance, like the routine that calculates the string length or the random number generator, which are sort of similar. They're under a lot of constraint. They don't change very much. But then there's a few things that change tremendously. And if you look at these things, they're in biology, we would call these under adaptive evolution. And if you think about what are these are things like the memory reroutine, the things that relate to the device raters. And these are the way that this is the operating system interfacing to the hardware or its environment. And then, this is the killer slide. It's a really bad slide, but it's a really interesting thing. So then, if you plot now connectivity versus constraint for the computer operating system routines, that's plotted here in this sort of strange nomenclature. But the key point is that as opposed to being inversely correlated, as opposed to being more connectivity associated with more constraint, here it goes the other way around. It's the opposite. It's the opposite trend. Okay? And this is very, very counterintuitive, but if you think about it for a second, it's very fun to think about. So why is that? Why do you have things that have higher connectivity actually have higher mutations in a man-made environment than what you observe in a natural environment? And you might say that doesn't make any sense. It makes a lot of sense that something that has more connectivity has more constraint. But let's think about it for a second. When changes are made in the computer operating system, they're made by man-made designers. And these designers believe that they can make a change without disrupting the thing. Okay? And then let's think about another network now. Think about the road network. If we had more connectivity as associated with more constraint in the road network, we would never have construction in Manhattan, right? Never, right? Because you would never modify a street in Manhattan, because all construction would happen in South Dakota because there's less connectivity there, right? But that's not true. So you obviously have a lot of construction in Manhattan. You have construction on bridges. And that's because the designers believe that they can make mutations and they can make changes without hurting the system. And so then their mindset becomes very different. Their mindset is that connectivity is associated with utility. So the more connectivity on something, the more used it is and the more important it is. And that's why you get a lot of construction on big bridges that lots of people go over, right? Because people use them and they actually want them improved. And you actually don't have that much construction in South Dakota because people aren't driving in South Dakota. And so it's a very different, and I think whereas in the natural system, it's just very different. You have random mutations, right, that just occur irrespectively. And so the natural system evolves in a very different way, right, to, given that you have a random as opposed to man-made design process or mutation process on it, you get a very different outcome. And so I find, myself, I find this very satisfying, because I find it that the analogy, I think, really helps you understand the result you get in the natural system. And so this is, I think of this as one of the powers of this kind of network kind of picture, where you can make a comparison. And the comparison gives you a lot of intuition and a lot of understanding for a system property.
So it's a variation on the rich gets richer, it's the bridge gets richer.
Yeah. Yes, that's right. Yes, no, that's exactly right. Yes, that's exactly right. It's a different. So then, just to summarize really quick, so what we did in the network section here was we talked about how we were going to take the linear annotation, okay, so, and reformulate that linear annotation into a network. We talked about how you do that. We talked about, the different mathematical things that we do in terms of taking the strongest edges, finding the distal edges. And the neat thing we found was that you could build a kind of hierarchical structure where you get most of the edges pointing downwards for the human transcriptional regulatory network. And the neat thing about this structure was that it, a lot of stuff kind of matches your intuitions. The transcription factors on the top they're better connected, they're more influential in terms of driving gene expression, there's information flow bottlenecks in the middle, and you have these little feed-forward loop structures that are created throughout the network. And then when we looked at the variation impact of the network, again, it kind of made sense. We had this general trend of more connectivity associated with more constraint, and in terms of the hierarchy, we had a little bit more constraint at the top of the hierarchy, which is a little more connected. And then we talked a little bit about how you can use kind of network analogies or cross-disciplinary analogies to give you intuition about what's going on. And so that was the point I was trying to illustrate about the networks.
Yes.
Yeah.
So, do we know where different class of transcript factors are in the network, like limit versus other general factors?
Well, no, that's interesting. So I didn't show that here. I mean, when we were building the network hierarchy in human, we did spend a while thinking about, there are particular types of factors, like for instance, fingers, or, I think it was, what was the other factor that we were quite keen on? There was another particular one that we were thinking about being on the top. I'm spacing on, I think it was a homeo protein or something like that we were thinking of being on the top. But we didn't find any obvious correlation between factor families or kind of high-level Go groups or something like that, that would tend to be on the top or at the bottom. And that was actually a little surprising. I would have expected that. We didn't found, maybe it could have been that one type of family of factors, you'd find helix loops, helix proteins would tend to be master controllers, and it's not like that at all. They're kind of fairly evenly distributed on the levels, and those divisions don't really map to this.
What about the diversity of the network versus in terms of constraints? If it's a highly diverse network, do you see any difference in constraints versus a low diversity network in terms of components, I guess is what they're called?
What do you mean by diversity?
So just more number of components. So if you have a network composed of 20,000 components versus a network composed of 2,000 components, I guess my thought is that in the network of 20,000 components is system-specific redundancy versus where in the network with lower number of components there's less diversity, and so changing a pretty good component will have a greater effect on the network in general.
I think that's probably true. It's unfortunately that type of analysis tends to get confounded very easily. So let me give you an example of what I mean. Someone might study one particular subnetwork or something like that, and they'll talk about the properties of this small network. But the truth is, of course, that small network is embedded in a bigger network that you're not seeing, right? And the question is, if you had all of the connectivity that existed, then, you know, say in two different organisms, and you made a comparison, this organism has a bigger network than this one, would you see that thing? And maybe you would, it makes a lot of sense. I don't think people have that. And the other issue is this sort of missing data issue. I mean, in a sense, the human network is bigger than, say, the yeast network. But is it really bigger? It's hard to answer that question. Like, is it bigger relative to the number of genes or number of proteins? And then we haven't completely filled in these networks yet. And so they're sort of relative to the amount of potential missing data. You know, but I think that's a very interesting question. I mean,
yeah, so the reason why I asked is because if you take this analogy and kind of expand it to the level of like an ecosystem, right? And so the analogous situation would be if you have a keystone species, which means that you have a highly connected component in the network. But the general idea in ecological systems is that if you have a large number of species in an ecological system, the stability is greater than if you have a system with lower number of species. And the idea behind that is what I said, system-specific redundancy.
you know, that's interesting. I think that's a great. I mean, I'm really interested in that, and that's exactly one of these interesting types of analogies. And people, I haven't personally worked on this, but I've seen there's a string of papers that people have written about kind of transcription factor complexity, say from E. coli to yeast to worm to human, and there's discussions about. I mean, this is sort of the issue again. Like, when you look at the scaling from like Mycoplasma genitalium up to, actually, we're not at the top of the level. Like maize is actually higher than us. Like a plant. Like we're actually like in the middle. Like a plant is, or like, you know, there's some. But if you look at the scaling through those, people do think a lot about, you know, what do you get with as you scale up? You know, is it that you get more redundancy? Is it that you get more duplication? And I actually think that's quite interesting. And I think it's you know, because I do think that I mean, a lot of biology scales to duplication. It's just very clear that a lot of times when you have double of something, what really is going on is you duplicate it. You know what I'm saying? And the duplication naturally gives rise to kind of you know, there's a neo-functionalization, and then there's a whole redundancy component. And the argument is maybe that's a good thing for the redundancy. I mean, but so it's I definitely am interested in that. And there's definitely people who write about that and have speculated about it. Yeah, it's interesting. Okay, so now what we've done is we've talked about models, we've talked about networks. And now I'm just gonna, oops, I went back when I went forward. No, same slide. We've talked about models, we talked about networks. And so now what I'm gonna do is talk a little bit about, I'm just going to walk through a few practical tools just to give you a flavor of some of the types of tool sets that people have developed. These are obviously a little more centered on tools that I've been involved with. And you've heard about some of these things, but I just think it's nice to kind of just quickly give you a snapshot of some practical tool sets. And then I thought I would close by just illustrating 2 points related to these tool sets. And I think you'll sort of see that. And I think that I'm hoping to get the things related to privacy and also the intersection of variant data with functional genomics data. So first I'm going to talk about chip-seq tool sets. And so I'm just going to basically have like a slide per tool. And just like a quick, you guys are going to go into a lot more detail on how these things work. I'm just going to say a few things. So one tool that is used to know about, or we are involved with, is this thing called peak-seq, which is a peak calling program. And just you guys are going to learn about this. You just basically look for peaks here, but it's a little bit more complicated because you just got to kind of think about your what you'd get without the transcription factor, or what you'd get in kind of a null situation. So this is what's called input DNA, and you can do some sort of simulation of this, you know, how you'd expect these peaks from this kind of null background, and score these observed peaks relative to that. And so this is what this program does, and defines peaks. Fairly simple idea. So we talked a lot about aggregation plots. And so this is a toolbox to create aggregation and correlation plots. So if you have these genomic signal tracks, you know, say for chip experiments, you know, it's useful. I mean, one thing you're not probably going to talk about as much, but there's a discipline of, I guess, signal processing that talks about, you know, how you process signals. And there is just some general things you can do with signals. And so one thing you can do with two signal tracks is you can correlate one track with another. We talked about that in relation to transcription factors. Another thing you can do is you can kind of correlate them about certain anchors, and that gives rise to the aggregation plots. And another thing you can do, which is kind of specialized analysis, but you can kind of, it's related to the correlation, but you can ask how much additional information do you get from an n+1 track relative to the previous n tracks, and that's kind of a saturation analysis. And so this is a tool that does that. And one of the things that's worthwhile to point out, this is an example of the correlation output. If you take the correlation of all factors with all factors, you get this nice correlation matrix, and you can do, now I'm going to kind of wave my hands and sort of describe a complicated calculation, but one very nice calculation to do is a principal components or spectral calculation on this data, which kind of gives you a very big overview of how all the things are working. And so this is just a pie plot of all the transcription factors interacting with each other in the genome. Then there's tool sets, of course, people have developed for analyzing networks, and so, you know, there's tools that will calculate things like the hubs, the betweenness, the bottlenecks, the little motifs. And there's a bunch of tools that do that. This is one tool we've developed. You probably also heard about Cytoscape, and Cytoscape is a very nice network visualization workbench that lots of things plug into, and this is a particular plugin into Cytoscape that does all these network calculations. I should just, you know, this is, one of the things about tools is when you develop tools, you spend a lot of time developing acronyms for tools, so that cool acronym. So this is one of my favorite tool acronyms. So the guy who developed this tool originally in my lab was this graduate student named Haiwan Yu. He's now an assistant professor at Cornell. And so he was just a very, very smart guy. He came from China, and he was just obsessed with Tom Cruise when he got here. So he decided to name his tool TopNet. I don't know if anyone understands what I'm saying, but this is like 10 years ago, so he called it TopNet. And then we had this, and I don't know if people know, but TopNet is a slightly militaristic kind of concept going on here. So then we had this other student who came in, and he didn't like the militaristic overtones of TopNet, and he didn't want to call his tool TopNet 2. So, but we wanted to relate it back to the original tool. So we spent probably a very long time coming up with an acronym. So we came up with this acronym, which I think is kind of cool, TYNA. It sounds like tRNA, but what it stands for, it's a recursive acronym, is TopNet-like Yale Network Analyzer. So we have the TopNet and everything's buried. So those are chip-seq tools, RNA-seq tools. There's tons of them. You're probably hearing a lot about them in this class. The RNA-seq, there's mapping tools. There's tools for building transcription expression. I'm going to mostly talk to you about kind of the bottom level of these RNA tools here. Again, most of the stuff that I've talked to you about is I don't want to say the higher level stuff, but I'll come to it a bit. It's a little removed from the actual reads. Okay, so there's a whole world of tool sets that, of course, map things quickly, and at the end I'll talk about one tool that really deals with the reads, this allele tool. But most of what I've talked about is really signal processing tools that deal with that signal. And it's important to think about that because I'll come to it in a second why it's important that you can just deal with the signal. Yeah.
So these things become very confusing. There are just too many of them, the tools like for calling, for mapping, for network. Are there like concepts like that, I think, or comments?
Well, no, I mean, that's an interesting question. I mean, it's kind of like, the Cambrian explosion and stuff like that. I mean, basically, when you had next generation sequencing developed, I mean, there's this profusion of people, everyone created it, a tool, they called it something seq. Like, we even thought of, we call one of our tools peak-seq. Then we started to think we have to call it the star-seq line of tools. That there's all possible seq tools, and so there was a tremendous number of tools people developed, and the idea is that hopefully the community is settling a little bit on some specific tools. And actually, one of the things that ENCODE really is pushing is not so much a standard set of tools, but coming up with standard quality control metrics, kind of standard ideas how to assess things and so forth. And that they've done that for chip-seq. For instance, one of the things that encode published, they published a chip-seq standards paper that talked a lot about the just a lot of metrics that you'd use to assess chip-seq. And I think the RNA-seq is a little bit more complicated than chip-seq. I think RNA-seq is a delta, more complicated. And so they haven't quite settled yet on, things to the same degree. But I suspect there'll be a little bit more. So in any case, there's a profusion of RNA-seq tools. Though I would say that to some degree it's dominated by the what do they call it, the tuxedo suite of programs, the bow tie, couplings and whatnot. But there's no real consensus, I would say, on tools. And so I'm just going to quickly show you a few tools and just kind of little different things they do. So now I'm just going to quickly say we have a suite called RC tools. It's not nearly as well known as couplings, but I'm going to tell you about something I want to introduce you. So I'm going to tell you about one thing it does later, which I think is really important, and when you see, you'll get the gist of the whole thing. Another tool. This is a tool developed by someone who really wanted to be intelligent, and so this one's called IQ-seq. And so another tool that people develop is transcript quantification in RNA-seq, where if you have a number of transcripts that are coming from the same locus, you want to figure out the relative abundance of them. And there's many tools that do this. This particular tool uses an expectation maximization approach. You're just basically looking for the most likely configuration of transcripts or isoforms from your genic locus. There's another thing people do with RNA-seq is they look for new transcripts, they also look for new regions that are transcribed in the genome. And so this is a tool called, this is another wonderful acronym I'll explain to you. It's called IncRNA. So I'll explain. The idea is that this tool is supposed to do integration of lots of pieces of evidence. And the one little vignette I want to get across here is that when you look at an individual experiment, particularly in a region that you don't really have as well characterized, a lot of times that one experiment doesn't tell you that much. But if you integrate many experiments, this is sort of an obvious truism, you can do a lot better. So this tool tries to find novel non-coding RNAs. And so let me just show you this one or two graphs. So if you take your known non-coding RNAs and you take, these are genes, say CBS, protein codings. If you look at the level of expression of these, this is a total RNA. This is a tiling rate experiment. It's a little bit old, but it would be about the same thing if you did a total RNA RNA-seq experiment. You can't really make a threshold to distinguish the red line from the blue line. It's just not going to work based on one. Like you could not pick a threshold so that only the known non-coding RNAs would be separated from the protein coding genes. Not going to work. But if you do, if you take two features, say you take the structure of your putative non-coding RNA and you take this result, you can actually separate a lot better. And if you take many, many features in a high-dimensional space, you can do fairly well. And so that's what this tool does. It finds non-coding RNAs. Now, this is this wins dumbest acronym of the year award. Because now this is my idea, actually. So we wanted to call it sort of integrating RNA, or IncRNA. But now anyone who looks at that knows that acronym looks a lot like lincRNA. So it's incredibly dumb. So when most people look at this, they think it's lincRNA. Really stupid, but in any case, you can learn a lot about how not to name things. So final RNA tool, this is just a walkthrough of all tools. So this is a specialized type of thing, but one thing that people want to do, particularly in a cancer context, is they want to find fusion transcripts. So if you have the genome rearranged, and you often want to find a gene that's kind of put together from two genes. That's called a fusion gene. And you can imagine you could use paired-end reads to find that. If you found that in your transcriptome you had a paired end that seemed to span two things that were really far apart and brought them together, lo and behold, you might think that could be a gene that was fused. Now, of course, to do this properly, you have to filter out lots and lots of potentially confounding things and so forth, mismapping and whatnot. But this tool tries to use paired ends to find fusion transcripts. And then after it finds a potential fusion transcript, what it does, which I think is quite useful, is it builds that transcript up and finds the potential place where the two parts of the genome came together. And now it has a new junction or breakpoint. And then it goes through all the additional reads in the experiment and sees if it can find that new splice or that new joining read as providing additional support. And this is called fusion seq. Okay, so that was just a quick run through of a bunch of tools. So now I want to just zoom in on two things. So I introduced the tools to you because I wanted to introduce this notion, which I think is kind of interesting, of, and this has been a theme through the whole talk, of the different levels of data. And so, and functional genomics sits in a very interesting way relative to these two different levels of data. So I talked about how the data is kind of arranged in a hierarchy. And I've given you this concept of there's the reads, there's the signal, and then there's kind of processed things on top of that, like the models, the networks, the gene expression levels. Now, one of the really important things, this doesn't come up so much in a functional genomics context, it comes up a lot in a genome sequencing context, is that the reads contain variant information, okay? And the variant information potentially is a very private thing. So people, I'm personally very interested in this privacy issue. So, historically, genomics has been a discipline about open data. Genome Project was one of the original open data projects, and in fact, the whole ethos of the 1990s, circa 1995 or 2000 Genome Project, was the competition between a private company, Celera, and this open public project. And this is really a big part of the ethos of genomics, is kind of open data. The genome should be free for all. Now, personal genomics, which is where the world is going, is a really different thing. This is a personal individual's genome. And it's a very private type of data. And so it's not really clear this data can be open. And, so what is the danger here? Let's just talk about this a little bit. So one issue that people talk a lot about is identification risk. Like, if you have your personal genome out there, someone can identify you from the genome. And that is an issue maybe you've heard about recently. There was this big issue of how some of the quote anonymous people in 1,000 Genomes were identified by this guy named Allelic, who figured out who they were. And that is a risk. Now, but then you might say, is that really a big risk? Well, they were identified. Does that really hurt them? Maybe, maybe not. But another risk that people don't talk about so much, but is also in the back of people's mind, is what you might call characterization risk. So imagine you even consent to be identified as putting your genome on the web. Does that mean that what risks are you open for? Well, it could be that people could mine your genome and find out bad things about you. They could find out that you have a psychotic murderer or something like that, or that maybe you're going to contract Huntington's disease, or maybe you're a bad insurance risk, or they're going to identify something about you, and that maybe you don't appreciate that, and so that there's a characterization risk there, and maybe they'll discriminate against you. Maybe they'll realize that, you thought that you were from this area of the world, but no, you're not. You're from a very different area, or something like that. So that this is an issue that people think about. Now, right now, if you go sequence your genome, realistically, it's not going to mean anything. You're going to get, your 6 billion bases, your diploid genome, and it's not. You're not going to make any sense of it, But the question is, if you put it up on the web, and 20 years go by, 50 years go by of biomedical research, will we be able to characterize genomes more? And we probably will be. And then the thing you also have to keep in mind, you might say, well, I don't care, because 50 years from now I'm going to be gone. But your children will be around, what I'm saying, and you pass on half of your genome to your kids. And so then this becomes a very kind of cosmic thing, in terms of like what the information is there. And so, there's a big debate about this. But there's also, at the same time, this idea that the whole promise of genomics is really through the mining of this data, putting all this data together and looking for large-scale correlations and large-scale patterns, and associations with phenotype, particularly disease phenotypes. That's where we hope to get beneficial things. So there's a kind of a yin and the yang here, and people are not sure what to do. So that's kind of genomics. But now, how does functional genomics fit in here? So functional genomics has an interesting little twist. So now, if you do a human genome resequence, it's all about the variant. I mean, it is about your variants. That's what it's about. But if you do a functional genomics experiment, are you really interested in the variants? Not necessarily. You might be, but you might not be. And so, often you're interested in just the biology of transcription, or you're interested in the amount of binding to a particular region of the genome, and the variants that flow along with that experiment are incidental collateral damage. And so on one hand, you kind of have decided that most functional genomics experiments, people are actually very happy with kind of the signal track, or sort of a simplification of the experiment. And only a small number of those experiments do people actually need the variant information. Okay? And it's important to realize, you might say, well, I'm doing a functional genomics experiment. I'm not really resequencing the genome. But please realize, if you do an RNA-seq experiment, for all practical purposes, an RNA-seq experiment, if it's fairly deeply sequenced, it's essentially an exome sequence for all practical purposes. And in chip-seq experiment, it's not quite there, but it's getting there. And so these are essentially potentially resequencings. So where does this all go? So one of the places it's going is, we've been thinking a lot about maybe there's some ways of organizing functional genomics data to kind of make them more easily available in a public way. And so the program I talked to you about, RC Tools, the important part of it is actually not the program, it's the file formats that it uses. And so we've thought a little bit about trying to create file formats that split the variant information away from the read mapping and the signal. Now, one thing you can do is you can just distribute the signal information itself, but that's a loss, because you don't have any concept of the reads anymore. But one idea you have is maybe you can actually talk about where the reads map, but not actually whether they carry a variant. And so that's what this format that I'll show you about, called MRF, does. And so it has the idea of kind of two linked files. One has the actual variants, and the other, it's just really simple, and the other has where the read maps. And you can separate the two in a very principled way and just give a person this first file, which gives them most of the information they need to interpret a functional genomics experiment without touching the variants, which is key. And this is just an example of some of these files. Basically all you're recording is where your read maps, okay, on the genome, the coordinates on where it maps. And if it gets into details, there's the read split. You have to take into account a split of the read. How does a paired end read map and stuff like that. When you drill into it, there's complexities. But the mindset is to create a kind of easy to read, simple textual format. And one of the really nice things about this format is it's also a tremendous compression. So if you look at this, say the raw output, this is from an ELAN file, this is an old file that used to come out of the sequencer, you'll get maybe a factor of 10 compression to build this MRF file, where you just store the variant and the mapping. And now, you maybe have heard of something called the CRAM file. So the BAM file, which you probably all, so you probably know that the raw format that people usually deal with from the sequencing is the FASTQ file. The BAM file contains the mapping with the reads. And then the CRAM is a compressed version. So now the MRF format that I'm talking about is actually very similar to the CRAM format. It's just, it's much more, in a principled way, talks about separating the two things. So that the key point I'm trying to get is that you could distribute a file from a functional genomic experiment that's easy to read, that's small, and that doesn't have anyone's private information. And that's the key idea. And I personally think this is going to be really important in the future, because I think that invariably we're going to do experiments on people. I mean, that's the reality. We're going to want to do functional genomic experiments on either human tissue or cell lines or something like that. And all of those are going to have the variants of a person. And whether you're interested in that person's variants or not, you're going to have literally millions of them in all of your experimental output. And so I do think it's important to think about how you can deal with them. Now, one thought process is all the data will be locked up or it will go into like dbGaP and so forth, but that is a big headache, and it's really nice to think about if there's ways of kind of doing this more artfully. So that was the main point here, just this idea that we can, most of the time we can get by with this MRF type of format. Now, what I'm going to talk about now is actually, as a funny segue, I'm going to talk about a particular type of calculation that you really need the reads for. You need the actual variant in the read for that calculation. This is a functional genomics calculation. I'm going to end on that. And these are actually, in some sense, the most interesting calculations, but I think that they really highlight kind of the difference between calculations that require variance versus that don't. Okay, so now I'm going to talk to you about some very specific calculations that involve what's called allele-specific effects. Okay, and allele obviously relates to variant. So here's what I mean. Let's take a chip-seq experiment. If you actually think about a chip-seq experiment, you have a transcription factor that binds DNA. Now, if you do it in a human or in a human cell line, there's actually 2 copies of everything there in the genome. There's the mom copy and the dad copy. And it could be, it doesn't have to be, but it could be that there is a sequence variant associated with them. And maybe that sequence variant is actually somehow, it's more not obvious, but somehow associated with differential binding. Well, lo and behold, your chip-seq experiment is going to read that out, because you're going to get a bunch of reads. And those reads will actually contain the variant that the thing is bound to. So in this particular case, you can see you bind much more to the T, the allele that has the T than the allele that has the C. Okay? So you can actually just directly measure the differential binding to those 2 different sequence variants. Now, that's simple and easy to understand. Now, it turns out this is about one of the most technically challenging processings to do. And why is it technically challenging? It's technically challenging because of this very complex effect called reference bias, which is a very, very confounding, tricky effect. And so the concept of reference bias is that imagine, okay, see, if you have reads from, say, me, and you map them back to the reference genome, and I have a SNP, okay, a heterozygous SNP at one position. For that heterozygous SNP, some of the reads are going to have the same sequence as the reference genome, others are going to be one different. So there's always going to be a preference to map against that read that's similar to the reference genome. And you're saying, like, who cares? It's a little bias. But that little bias will completely skew the whole effect. So you have to correct for the bias. And so let me explain to you a little bit about how you measure the bias and how you correct for it. So imagine if you were looking at a site in the genome where there was no allelic effect. The maternal allele and the paternal allele were transcribed at the same level, bound at the same level. Well, if you looked at the amount of maternal versus paternal allele you get, you'd expect to follow a binomial distribution. Coin flip, you know. Sometimes you'll get a little more maternal than paternal, and sometimes more paternal than maternal, and it'll follow binomial, you know, centered at 50-50. And just like this red graph, okay? Nice and symmetrical. Now, if you do an allelic experiment, what will happen is you'll get a pileup. You'll get lots of certain types of situations were really dominated by one variant versus the other. But relative to the reference genome is completely arbitrary. It has nothing to do with maternal or paternal. It's an arbitrary thing. So you expect that the the allelic experiment will show differences at the sides, but that they should be symmetrical. Now, if you see an experiment where all the time the reference allele has a slightly higher level of binding or a higher level of expression, the implication is that's reference bias. So this picture shows reference bias. Okay? Now, how do you correct for reference bias? Well, the way you correct for reference bias is kind of the way that you'll probably be doing all functional genomics experiments in the 5 years from now. You don't map your functional genomics experiment to the reference genome, because the reads didn't come from the reference genome, right? The reference genome is an arbitrary construction. You map the reads from your functional genomic experiment onto the personal genome of the individual that you sequenced or that you did the experiment on. So basically, the simple minded way of correcting is you build a personal genome. And so in this particular approach, what you do is you take the variant calls for a personal genome and you instantiate the maternal and the paternal haplotype. Now that's not a trivial matter at all, because it requires having all the variant calls. It requires having them phased, so you know which is maternal and which is paternal, and then building those two genomes and then mapping against those two. Fair enough, but in any case, we've developed a way of doing this. And then once you do that, we have a little tool set that we call the Allele-specific pipeline, where you map against the maternal and paternal genomes, and then you basically do some careful statistics to look for, do you now have more maternal than paternal reads at a given heterozygous SNP? And if you do, you compare them against a binomial expectation, and then you say you have allelic imbalance if you're greater than this. Now, you have to be a little careful to correct for multiple hypothesis testing, so there's a little bit of simulation. And the other thing that you have to do that's tricky too is, in addition to having this is why it gets complicated, in addition to having a maternal and paternal genome, you actually have to have a maternal and paternal annotation file, right? Because remember, the gene annotation of your mom might be slightly different than your dad. The mom might have a slightly different SNP junction and this and that versus your dad. So you actually have to have two copies of your gene sets, and you have to map your, say, transcriptome read perfectly to this. It's very kind of a compulsive process. You have to carry it all the way through. And so I'm going to show you what happens when you do this. So the test case that everyone does everything on is this thing called GM12878, which is, or NA12878, which is this individual that was incredibly deeply sequenced as part of 1,000 genomes. And has also is one of the centerpieces of ENCODE. And so in just a kind of a specific analysis, this is a number of factors, sequenced a few 100 million reads. This is an early experiment. And, a fairly deeply sequenced RNA-seq experiment with maybe 150 million mapped reads. So when you do this, you can map your reads to the reference genome, you can map them to the paternal genome, or you can map them to the maternal genome. And now what happens when you do the mapping, the first thing you can do, before you even do the calls, you can ask, are there any reads that just change positions between the reference genome to the paternal genome to the maternal genome, okay? Do they stay in the same place, or do they bounce around, okay? And you just tabulate them. Now, the thing that gets really weird, and this is why this stuff gets complex, is you can have a situation where this read here, these reads will say map in the same position here, but this read will move to another place in the paternal genome, not because there's a SNP here that's affecting anything, but there's a SNP someplace else in the genome, way far away, that's sucking it up. So even not at the place that you're actually looking at, but all over the genome, there might be a new duplication involved, there might be some additional thing that will subtly change the mapping at a given site. So a very global effect. So you can't just say, cut out the region that you're interested in, see if you map better to the maternal or paternal. You have to look at the entire genome and then consider the whole mapping as a whole. And when you do that, what do you find? Well, you find a very small effect. You find that basically, I won't walk you through these numbers in detail, but if you look at the number of reads that bounce around, you're talking about 0.1%. Really small number of reads. You might say, who cares? Doesn't make any difference. But that very small number of reads that bounce around, those are the reads that are going to give you those are the ones that are really the ones that you care about, because those are the ones that are sensitive to the sequence differences between mom and dad. And when you do it, when you do your little picture, this is what I mean by correcting. You see, this picture has reference bias, and it's subtle, but this picture doesn't. It's flattened to that. Okay. And now what happens when you what are the numbers that you find? Well, you find after you correct for reference bias, do all your testing right and all that, you find that about 20% of the genes in the genome have allelic expression. Now, this is a complicated calculation because it's not 20% of all genes, it's 20% of the genes that have a heterozygous SNP that you can actually use to assess the allelic effect. Not all the genes. Okay. And you find about the same fraction of non-coding RNAs that are accessible, or allelic, about a fifth. And then you can try to put the binding together with the expression, and here's an example of this. You know, here's a gene where you have allelically expressed exons, and you can also see it's associated with allelically expressed binding of polymerase. And you know, you can see the paternal and the maternal is very different. And you can, you know, overall you see about a fifth and a factor of about a fifth difference between maternal and paternal binding. And then what you can do is you can build a network. You can build a network of your allelic. So if you have a bunch of factors, now I have a bunch of factors here, I'm going to build a network where what I'm going to do is I'm going to color an edge if the edge only exists in the mom or only exists in the dad. So I have my whole big regulatory network, but I can actually construct a maternal specific or a paternal specific network. So that's the network that just has the edges that exist in mom or just the edges that exist in dad. Not that many edges here, but still it's quite interesting. And then I can ask if I have a factor and it regulates this gene, mostly red is maternal, blue is paternal. If it regulates a gene mostly maternally, is the gene mostly transcribed maternally or paternally? So I color the nodes by if they have ASE. Right? So the lines are ASB, the nodes are ASE. And you can kind of see in the picture that you tend to get a very kind of consistency effect. Like, you know, if you tend to have red lines going into a node, not always, it usually tends to be red. So mostly when you have, if you have allele-specific binding maternally, it's associated with allele-specific expression, that part of the chromosome's on. Not always. You see here, for instance, let's find this example here, one where you have a blue line going into a red node. Not always. But what's neat is when you take, this is a small number of transcription factors, when you look at the entire ENCODE compendium, and you look at examples where you get more and more binding on something that's either maternally specific or paternally specific, as you get more and more binding, it becomes much more unified. And so you get these kind of clumps in the regulatory network where it's all maternal binding, all paternal binding associated with maternal or paternal expression. So it's kind of a neat thing. And then the final thing you can do, which is kind of neat, is you can go to the sites in the genome that have allelic activity, that means binding or expression, and you can do your selection calculation. And this is I'm not going to explain how this calculation is done, but this is a skew in the allele frequency spectra calculation. And what you find here, which is actually maybe counterintuitive, is you find that the sites that are allelic, that means they differ between mom and dad, actually are under less selection than the non-allelic ones. Not completely intuitive. So in any case, we have a little bit of it. We have a website associated with this, Allele-seq, and you can download all the allelic genes, the allelic sites, and more importantly, you can download the tools that build the personal genome and do the calculation and stuff like that. Okay, so I'm going to summarize now what we've talked about. I've talked to you about 3 things: the building models, building networks, and then at the end I gave you a quick walk through some tools, and I wanted to just highlight 2 aspects of tool sets. One aspect is this privacy aspect and how we really have to think, particularly for functional genomics data, about how we can separate the signal parts of the data from the variants. And then I talked to you about at the end in detail about a calculation where really we're interrelating the variance in a functional genomic experiment with, the signal in a functional genomic experiment with the variance. And this is an allelic calculation, which is very interesting, and we see the differences between the different sequences. And so I just, I'm going to acknowledge lots of people involved in this. And so the models stuff, these are all the people involved in the models paper, and is probably obvious. I've highlighted in this one main person over and over again, Chao Cheng, who was a postdoc in my lab. He's now an assistant professor at Dartmouth. He developed a lot of the statistical model machine making. He developed it in many specific methods papers, and then he eventually applied it into the bigger encode world and stuff like that. And he worked a bit with Kun-Kyu Yan, who's associate research scientist in my lab. Now, the regulatory network stuff I talked to you about was mostly the encode presentation of the human regulatory network. And this is very much the formal encode collaboration thing. So there's hundreds of people in encode. This is a subgroup of about 50, 60 people. It's called the networks element subgroup. The networks element subgroup is listed here. Oops, somehow, well, my pointer is not pointing, but in any case, it's listed here. And the group is kind of a fusion of these are the data analysts, and these are the people that generated all the data. In particular, most of the data was generated from the labs of Mike Snyder and Rick Myers. They generated all the hundreds of chip-seq experiments. And the main analysts include Anshul Kundaje, who's just started as a faculty member at Stanford, Chao Cheng, who I just told you about, Kun-Kyu Yan, Jasmine Yu, who was a student with me, now she's moved on to a postdoc, Joe Rozovsky, who's associate research scientist in my lab, and some other individuals. So that's the network stuff, and then the tools acknowledgement. There's just a lot of people here, so these are just different little tool sets involved. You can see Joel was involved in the peek-seq tool. The privacy stuff in the MRF is really this Lucas Habeger and Andreas Sponer, and also Doug Greenbaum developed that framework. The Andrea also developed the fusion-seq tool with Mark Rubin at Cornell, and the Allele-seq tool and so forth has really been spearheaded by Joe Rozovsky and Alexei Abazov. And the networks tool, TYNA, was spearheaded by Kevin Yip, who used to be a student, now he's a professor in Hong. And with that, I'll thank you for your attention.
So I wanted to follow up on the de-identification stuff, which is intriguing. I was thinking that, even if you have a chip profile, you may still get information about maybe gain or loss of a binding site or a factor, or maybe from a global pattern, infer something about the activity of a particular pathway or whatever, right? But then again, that data is for a particular transcription factor, which is much more specific than the genome sequence. So do you have a sense of how much of the information are you hiding? And and also, following up, is it a fundamentally unsolvable problem that if you have any kind of functionality in any representation, if it contains information that's specific to the individual, right? This is always going to carry this risk with it.
Well, no, that's a good question. I mean, so for instance, some people believe that even more than a chip experiment, RNA-seq experiment has, even if you look at the gene expression levels, they have the trace of the person. Eric Shad in particular believes this. I don't know if I agree with it. I mean, so one view is for instance, if you have a gene deletion, well, the gene's not expressed, fine. But the problem is that it's so complex to answer that question really well, because take gene expression. If you take one of my cells, one of your cells, they're going to be different. They're going to have different expression. But do those different expressions, do they reflect slight differences in cell type, or are they the same cells? And then do they reflect differences in condition? Maybe it's warmer over here than over. I mean, just even a slight difference in condition will give rise to a difference in gene expression. And is that difference a reflection of my variance, or is it a reflection of the condition? And I don't know if anyone's really can clearly say the differences that they observe between intra-individual gene expression patterns completely reflect variance. Now, formally, I've been thinking a lot about this. There are a lot of experiments that people do, you probably know about, where it's called the EQTL experiment, where they try to correlate, and also the allelic things, where they try to correlate differences with variance. Now, you might think, well, you could look at it that way. There's not that many EQTLs you can, statistically supported EQTLs you can get from an experiment. And it's a lot less than the number of variants you can get from it. So the average person has 3 million variants, okay? Now, in terms of unique variants, it's a very complicated thing to answer, how many unique variants a person really has. But certainly, if you think about it in the framework of a chip, you have maybe a tenth the number of, like if you do a SNP chip on you, which is, pretty good proxy for your genotype, that's about a tenth, okay? And I mean, you know, so a tenth of 3 million is still a lot larger than the number of EQTLs that you could maybe back infer from, you know. So I'm interested in this, you can tell.
That's interesting. So it seems like you say all these other data, they have sources of variability that confound the analysis, but the genome is the only thing that doesn't depend on external conditions. Right?
Well, no, that's true. No, that's true. I mean, that's the issue with functional genomics. I mean, you know, I mean, certainly in functional genomics, we're going to be interested in relating the functional genomic signal to genotype. But there's a lot more stuff that the functional genomics depends on. There's just so many other things. And maybe we want to study those other things and not get into this whole muck of, you know, the genome variants.
So on the same issue, I mean, there must be portions of the genome that make it easier or less easy to identify individuals, right? So, I mean, there was that paper that was published showing that through some method, to be honest, I haven't read the paper, but through some method they were able to identify some people, right? And so why can't, I mean, so if you sequence someone's genome at really low coverage, some variants would be basically non-resolvable, right? And so why isn't it the case that you can be like, okay, there are, you know, 300 regions of the genome that we know can very well help us identify people. Why can't we just basically remove reads? And so the call is still there, but it's too ambiguous to really make a confident call, and therefore you have some ambiguity, non-resolvable ambiguity to kind of identify individual people.
No, well, that's an interesting idea. I mean, just to be blunt, that's just not going to work, and I'll tell you why it's not going to work. So it's interesting. So first of all, there are loci that people definitely use for identification. For instance, a very famous group of loci called the CODIS loci. Those are the loci the FBI uses to identify you. It's like the number of like short terminal, short tandem repeats or something. But the truth is that many of the regions of the genome, there's many. It's not like there's a few regions of the genome that can identify you. Your identifiability is spread over your entire genome. So you could cut out. I mean, I could cut out 90% of your genome. I could still identify you. And I could cut out 90% in many different ways. And then there's this other issue that is really a very complex issue, is that, and that makes this all tricky, is that there's this well-known issue of linkages, LD, linkages equilibrium. And what LD means is that and this gets complex. So what LD means is, if I tell you about the SNPs over here, they're linked to the SNPs over here. So there's a very famous case, for instance, happened right here, of Jim Watson. You know, he had his genome sequenced. And he was like, well, I don't want you to know my ApoE phenotype or my ApoE genotype because, you know, ApoE is associated with Alzheimer's, and he didn't want it. So he said, I don't want my ApoE to be able to leak. But I mean, Jim Watson, you know, is a mega genius, but that was incredibly stupid because it's trivially easy to figure out his ApoE genotype from the neighboring SNPs around there in LD. And so someone wrote a paper where they're like, well, I can figure it out anyway. You know what I'm saying? And so the point is, it's very, very tricky because of the correlative structure within the genome and also the correlative structure of everything to actually hide things. You know, you think you're hiding something, but you're not really. And so, you know, one extreme of that is you can't hide anything. You know what I'm saying? And that everything has to be, you know, locked down and so forth. But that's it. You know, I'm actually of the privacy mindset. I tend to believe in privacy. But, you know, if you take that viewpoint, it's a tremendous headache for research. I mean, like, for instance, you're going to do some class right now, presumably. You can't sit there on a computer and just, you know, dial up your sequence. You know, you have to be consented to do that. You have to go through proper authorization. You can't, a lot of the operations that you just take for granted as trivial suddenly become major amounts of work. And, you know, it's not a good world to be in. So,
I agree in principle with the notion of trying to protect genomic information, but on the other hand, I feel that the information that insurance companies have about our family history, whether or not somebody has diabetes, whether or not somebody has smoked in their life, that kind of information, at this point at least, is so much stronger in terms of predicting anything. Not to mention all the pictures of drunk people on Facebook and stuff like that, that, you know, potentially could be used against you in some way. So I'm a little skeptical that I think we're going in one direction, trying to protect genomic information, in a very different direction in terms of what insurance companies have about us, how many pictures of us are online, and all these other things that potentially could be just as harmful
Well, no, that's completely correct, and completely correct. And yes, I mean, right now, right today, if you sequence my genome, it actually, if I put my picture of myself on the web and I put my genome on the web, my picture reveals much more about my face than my genome. I mean, my genome reveals almost nothing. On the other, but the problem with the genome, the fundamental problem is that 50 years from now, that might not be true. And the question is, you know, I have 2 daughters, right? And so that's where it gets cosmic. You know what I'm saying? Like, I mean, would you, I mean, like, you know, for instance, I'm very friendly with some of the scientists who sequence themselves, like, you know, George Church or Mike Snyder or whatnot. They don't care. They just really don't care. But, you know, the thing is, first of all, maybe they don't care. But what about their kids? I mean, can they make that decision? Even if, can they even responsibly make that decision for their kids? Okay? That's not clear. They even do that lawfully or legally, right? And, you know, and then I think that's where it gets really weird, you know, and to think about. And that's not the same as your medical, I mean, I don't think your Facebook profile is as heritable as your genome. Your genome is really heritable. I mean, you know.
Well, if there's no more questions, let's thank Mark again.